So my name is Premi Haynes, and I am one of the board members of Friends of FSH Research. And thank you everyone for coming here for this research update meeting, which is right before the auction. So people who are in person will get to go to the auction as well. And online folks, thank you so much for joining. I know you're from different places and time zones are different. So I'm really glad that you're able to make it here. This first slide I just wanted to show is the 2024 funding recipients. So these are the people, researchers who got funding from our organization in 2024. And the auction itself is one of our biggest funding that we get is from the auction. So we really appreciate all the donors who fund FSHD research here. And we have been doing this since 2005. And so we have funded more than $8 million for FSHD research. So thank you so much. Some of these researchers who we have funded are, I think, here, and maybe you can raise your hands if you're here. Hi. Thank you for all the hard work that you're doing. It is a difficult disease to study and research, and we have come a long way. So appreciate all your help and hard work. Okay, now I'm going to turn this over to Seth. Seth is going to moderate this event today, and he has put a really nice program together. And there is a great lineup of FSHD experts and presenters. So thank you.
Thanks so much, Premi. Thanks to Premi and Amanda for getting this all together. It's one of the biggest blessings when you're trying to get something like this going, and the email response, I think, with Friends of FSH is still, on par with that. So I'm really happy to be here today. I think this is such a nice forum for me to be in. We're so used to kind of presenting it sometimes at scientific meetings where you don't get this sort of informal quality. And so I'm really excited that, for the presenters that are here, I think we're all trying to give you a sense of where the field is in the current day with MRI. We've made a lot of progress since I presented last year, and I think you'll see from the presentations today that we have just made an enormous, enormous sea change in how we can think about these biomarkers to understand the disease that's just highly heterogeneous. So we kind of have been very tool limited for a lot of years, and I think we've overcome that now. And it's super exciting to see what's coming out of all these different labs. And I think with help from our colleagues in Florida who have done an enormous amount of work with DMD, kind of that's been in this trial space for much longer. And with the trials that are now happening in FSHD, we really have the opportunity kind of today decide like what's the roadmap forward. And so I'm, of course, very biased. Like I love these MRI biomarkers. And I think that there's a lot of reason to think that this is a super helpful tool in how we're going to understand and follow FSHD. So thank you all for being here. For everyone online, we look forward to your questions. We will try and get through a couple questions at the end of each speaker. We probably only have time for 1 or 2 questions, and we'll try and deal with a lot of the stuff in the panel discussion at the end. If we don't get a chance to answer your questions, we will collate all those and try and answer them in some way. So rest assured that if you have a comment, please put it in the chat. We won't be unmuting people during the presentation. So, but anyway, thank you all for being here. It's really it's a real treat to get to have this discussion today. So our first speaker will be Doris Leung. Doris is our amazing colleague from Johns Hopkins. And owing to the informality of this, I thought I would tell a little secret about each of the presenters that maybe other people don't know. And so one of the things that I've gotten to know Doris a lot over the last few years that we've been working on this data lake project that was funded from Friends. And oftentimes now with ChatGPT, many of you in the room are probably thinking, I have a 10,000 word document and I need to reduce it to 5,000 words really efficiently. So you can try ChatGPT. I just send my stuff to Doris because Doris has a really amazing ability to kind of simplify things down in a way that is amazingly helpful. And she's just a really wonderful collaborator in this whole process. So we're going to have a little bit of a live presenter, online presenter kind of flip-flop. And so we're going to see how this goes. So Doris is in Baltimore. And so I'm going to stop sharing here and I'm going to let her take over.
All right, thank you so much for that introduction. And I'm just going to go ahead and start sharing my screen. So thank you so much for inviting me to speak today. My name is Doris Leung. I'm the director for the Center of Genetic Muscle Disorders at the Kennedy Krieger Institute here in Baltimore. And I am the director of a multidisciplinary clinic for patients with all types of genetic muscle disease. I'm also the site PI for multiple clinical trials in muscular dystrophy. And so this list of disclosures are all sponsors of clinical trials and natural history studies that we've been conducting up here in Baltimore. So today I'm going to be presenting some data from a cohort of FSHD patients that I've followed for just under 2 years using whole body MRI. This topic I hope will help us set up the other talks in this session that will go into much greater detail about the process we're going through in developing MRI-based measurements as a biomarker or a surrogate outcome measure for clinical trials in muscular dystrophy. So when I start talking about clinical trials, I think that the thing I always emphasize to my students and to my trainees and to my patients is that clinical trials are aiming to improve muscle function. And this seems like a very, very obvious thing, right? But I think that, when we get sort of bogged down in really deep to these MRI studies, right, we can sometimes forget that the MRI doesn't necessarily directly measure muscle function, which is really the clinical benefit that we're hoping to get from treatment in muscular dystrophy. So this figure is from a survey of over 1,100 European patients with FSHD who were asked about the symptoms that most affect their daily lives. And so you can see that the symptoms that were most frequently cited really do center around function, either of the arms and the hands, but also in walking and in ambulation. So knowing this, ideally, what we would be using in these clinical trials are outcome measures that are entirely based in muscle function, like how fast do you walk? And when we've tried to do this directly, we've come to a lot of challenges. And so when we follow a large group of patients, like the MOVE cohort, for instance, we will observe that there is a gradual decline in function across the group over time. And this has been replicated in a number of different studies. But here's a figure from my own FSHD cohort where I've tracked the distance that they walk in 2 minutes over the course of 5 visits that were spread out over 21 months. And so you can sort of see like with each different colored line is a different patient, and you can start to see the issue here, right? That there's a huge amount of variability both within each patient and also between each patient. And so and this is not surprising when you consider, right, that actions like walking involve the coordination of over 200 muscles. And it can be influenced by a lot of other factors, such as fatigue, right, that can vary day to day, but are not necessarily directly related to disease severity at that time. And so imaging, I think, can really help us address some of the limitations that we've encountered in directly trying to measure function. So this is the demographic breakdown of this group of patients that I recruited through my study. It's a relatively small group of 30 patients. And the study was actually funded by the NIH through a K-23 Career Development Award. And so one of the more unique features, I think, is that I didn't place any restrictions based on strength or muscle function. And the idea I was going for was that, scanning can capture pretty much any stage of disease, even patients who are asymptomatic, patients who have lost ambulation. And so as a result, we actually have a very wide spectrum of disease severity presented here. And when we start to look at their scans, right, one of the points that I think really always stands out is that in FSHD, disease is happening very much on a muscle by muscle basis. Okay, so if this is the cross section of the thighs of a patient with fairly early disease, and so you can see, just to orient everybody, there's the bone is the bright spot in the middle, which is the femur. And then the quadriceps are kind of make up the top half, and then the bottom half are made up of both hamstrings as well as adductors of the thigh. And you can start to see that, this is a pretty normal looking MRI where the muscle appears dark, the subcutaneous fat appears bright. But if I pointed you to this muscle here and its corresponding muscle here, you would start to see that this is beginning to look a little bit abnormal. This muscle is the semimembranosus, and you can see that this muscle is starting to become infiltrated with fat and fibrosis, even as all of the other muscles are essentially normal. And then in this patient in the bottom panel, who is much more advanced, you can see that the hamstrings themselves are very advanced, and so have been almost completely replaced by fat and fibrosis. But the quadriceps are actually still hanging in there, with the exception of, you can see that there's this notch, right? Where a single muscle called the rectus femoris is supposed to be. And so in these types of patterns, we will start to see over and over again among patients with FSHD. We can also follow the progress of a single muscle over time. And so this is an example from one of the patients in my cohort. And we kind of we program the scanner to look at the MRI in a couple of different ways. And so when we look at STIR, it's a specific type of sequence that causes free water or inflammation to be extremely bright. And you can, and I already put arrows here for this muscle. You can see that this one muscle called the semitendinosus, which is also a hamstring, is very, very bright at baseline. And as this patient is scanned over and over and over, over the 21 months of the study, this brightness tends to go away. And so it is believed that when we go into and look at these muscles, this represents a more active stage of disease. When our colleagues have biopsied these muscles, they have found higher expression levels of DUX4 during this time. So the corresponding scans that show muscle versus fat will also kind of reflect change over time. In that, here's the first page, the first scan shows that it's not a normal appearing muscle, but as we advance through the whole course of the study, much of this tendinosus muscle tends to disappear. So one of the more straightforward biomarkers that has resulted from the different MRI studies that are being done um, is this fat fraction concept, where the volume of the muscle is increasingly fat replaced, and this is called the fat fraction. So after I had collected all of these scans, we partnered with Seth Friedman and his colleagues at Seattle Children's, who connected us with Springbok and got some funding from the Friends of FSHD Research to use their artificial intelligence algorithms to try to quantify muscle and fat volumes in our whole body scans. And so this is just a very small chunk of the data. And these are fat fractions that have been measured in the hamstring muscles as well as the quadriceps muscles across the just at base in my group. And so each of the blue dots is the fat fraction of a single muscle. So each one of these bars, each one of these muscles should have about 30 dots in there. And then the yellow bands are actually where the median is. And so you can start to see one of the challenges that we're going to be facing when we are measuring muscle disease and trying to map that back to function. When we're measuring the strength of, say, the hamstrings muscle, it's measuring the strength of multiple of like all four of these, the relative contributions of four different muscles, and all of them can be at different stages of fat infiltration. So, and kind of to build upon that, another challenge is that even though there are some muscles that are being more rapidly replaced, this is a relative minority. And so you don't need to pay attention too much to the individual numbers of this slide. But kind of the point I wanted to illustrate is that this is the average change in the fat fraction over 3 months. And so here's the mean and the standard deviation. And here is the change over 21 months from the beginning to the end of the cohort. And so here we have just the leg muscles. And so, 52 muscles were segmented in the legs across 30 participants. Oh, excuse me. Which means that we followed 1,560 leg muscles over time. And so when we scan in a very, very short term, we're seeing very, very small percentage changes, like essentially close to zero. When we start to look at the 21-month data, we're starting to see that this number is creeping up. And that we're on the average, we're increasing by a couple of percentage points over this period of time. But the standard deviations are extremely wide, which really signify that there is change, that the fat fraction is gradually increasing, but at very different rates across the population. And just these last 3 columns here over time are kind of giving us an idea of sort of like what is the rate of change. And so over 21 months, about sort of 23% of all of the muscles that we scanned increased in their fat fraction by more than 3%. About 13% increased by actually more than 5%. And then only about 4% of all of the muscles that we scan increased by more than 10%, which is like the patient that I showed earlier in our patient with her summary tendinosis that we just happened to catch at the point at which the replacement was accelerating. So this is, we can more or less the next big hurdle I think that we are facing when we are trying to develop imaging biomarkers and mapping them back onto function. I think the next talks will go into a lot greater detail about how we're seeking to understand the relevant features of individual muscles, how we are relating and identifying the associations of these muscle specific features back onto muscle strength and function, and doing this in a way that will generate usable biomarkers and surrogate outcome measures for clinical trials in this population. So this is my last slide. I wanted to thank all of our partners, particularly our friends of FSHD and the NINDS for funding the research that I presented, and also to the FSHD Society, who really made a huge effort in helping us recruit patients from all over the country to be in this in this study. So this is my last slide. I have the chat open, and I will go ahead and stop sharing, I think. And I'd be happy to answer any questions.
Hi, Doris. My name is Laura. Apologies, I don't have my camera on. I'm based in Sydney.
Hello.
And hello, fantastic talk. Thank you. I wondered when you're looking at these individual muscles, 1,560 muscles across the 30 patients, you identified a lot of variability within a patient, between patients, with respect to fat content at the individual muscle level, using the Springbok analytics tool. Wondered if you'd looked at the actual, other biomarkers like muscle cell biomarkers in those patients at all that could, or even blood biomarkers that could account for that variability.
So for this particular study, we did not collect blood biomarkers, and really, not for any particular reason that we don't think that we were going to find anything. It was entirely related to cost, in that people are actually floored to discover that I was actually able to fund this entire trial on the budget of a K-23 award to begin with. And so it basically paid for the scanning, and particularly for some transportation costs for the patients that participated. But there really was not bandwidth to follow sort of biologic biomarkers as well.
Understood. Thank you. Do you have a hypothesis to why you're seeing that variability? 21 months time frame seems like a very slow change. Do you have a hypothesis why you're seeing so much variability within the patient?
I think that there's a couple of different things, and I honestly think that we have, well, I'll start this again. Okay. I think that there are a lot of factors that go into the severity of disease. Okay. Some of which we have identified. Some of them are genetic, such as the size of the deletion, right? And in others we have yet to discover. But I think the probably where the reason why I think that we're seeing a lot of variability that in this population is that we are scanning them just in this 21-month window of their lives. And so so a lot of it is actually chance, that in other periods of their lives, had we picked a different 21-month period, we could have been seeing more or less progression at any given time. It's just, it's clearly um, a widely variable cohort of people to begin with, with my youngest patient being like 17 and the oldest patient being 75. I think it might have actually surprised me more if there had been less variability that we ended up seeing.
Thank you.
I'm sorry.
Yes,
Thank you.
please.
Robin Fitzsimons is also from Sydney. Just a comment on the variability. I have deja vu with the early multiple sclerosis trials, and of course, MS is also a disease with, to a very significant extent, variable and unpredictable progress. And the early trials, I recall, were beset by statistical issues related to this variability of progress, incidentally also an inflammatory disease. And it goes back, in fact, to Landouzy and Desjardins, who described FSHD as, excuse my poor French pronunciation, sa marche capricieuse et irrégulière, the capricious and irregular progress, in contrast to other diseases. So it's not surprising that there will be difficulties in interpreting at relatively short periods of clinical trials.
Thank you. Thank you very much.
Thanks very much for those questions online. If you wouldn't mind submitting them in the chat, it'll help us in the room a little bit to coordinate the folks in the room. So we just have one more question from Stuart in the room.
Yes, so you use the SIR positive as your baseline. So I presume that's because that's a good point for seeing rapid change. And the second part question is, once you hit SIR positive, is that the point of no return, or do you think like an intervention, a DUX4 inhibitor could still rescue the muscle after that point?
So Doris, the question is whether you think STIR positivity is sort of a good marker of a muscle's rapid change, and then whether it's still something that can be modulated by some kind of a DUX4 targeted treatment.
Right. Okay. So I think one thing I did want to sort of clarify is that, we do not, I think we just, I scanned like many patients to find, this one muscle that was kind of STIR positive at baseline and then showed progression. I think, we still have a lot of work to kind of see how does STIR actually progress, right? And how long will muscle stay STIR positive? And I think that what we're discovering, not necessarily in this cohort, but in other cohorts, is that it's a lot more complicated. That the body's relationship with STIR is a lot more complicated than we thought. In that even among the muscles that are not STIR positive, we can still see progression of fat infiltration, albeit maybe not to this, not to the to this very extreme extent. And so I think that, I think that the STIR positivity does represent something. It does represent this more, kind of that somehow there's the triggering of this cascade event that is extremely myotoxic. That being said, I don't think that it explains everything. In which that would, and it would not explain everything that we even observe in following these patients as well.
Thank you so much, Doris.
Oh, you're welcome. Thanks for inviting me.
If there are other questions, just please put them in the chat, and then we'll try and wrap them all up towards the end. Thank you all so much. Great questions. And I hope I'm going to answer at least some of them in this talk. And then we're going to take you sort of, I think, down this little odyssey of how you take this big giant spreadsheet of numbers and you turn it into something that is pretty understandable by the time Silvia gets to her talk. So that's my hope. Here are my disclosures. Okay, so I'm going to try and answer one pretty simple question today, and then I'm going to give you a little preview of what we're doing with the pigs. So the really simple question I'm going to try and answer is that forever, since we started these studies where we were doing MRI and biopsies, we wanted to try and understand which of these variables or which of these combination of variables predict change over a year in fat. And so is it the RNA measures that we're getting from the tissue samples? Is it pathology? So that's what I'm going to try and answer in the first part of my talk. And then I'm going to give you kind of a preview of what's happening with the pig atlas and the pig studies that will definitely take the next steps And, if everyone would love my punny picture and my next steps, that's kind of what I'm going for here. Okay, so how do we get here? So I got my first grant from Friends of FSH about almost 20 years ago now. And at that time, we started to collect some data. Some of you in this room are probably in the scanner. George, I know you're here. So we started to collect some data. And over time, we got a Wellstone Award, and then we got another Wellstone Award and another Wellstone Award, and we ended up with all this data. And in 2022, I sort of had the problem we always have, which is, okay, time to write some more papers, get some graduate students. But we were really only analyzing like little sections of the muscle. And so I found this paper by Silvia and her group that came up with this really cool AI method to basically shrink wrap the muscles, which you can imagine is kind of what we're trying to get our eyes to do. And I thought, this might be the solution, will they collaborate with me? And then I realized that, oh, wait, this is University of Virginia. I went to University of Virginia back in the day. And so I reached out to Silvia and Springbok, and we've had this just amazing collaboration over the last 2-ish years. And what you're going to see is like, this is where we were 2 years ago. And sort of what you'll see in the next couple talks is what we've learned today. So these are the kind of metrics we can now make. So we were just obviously measuring single slices, but now we can create these really amazing surfaces that can be mapped by different parameters like fat fraction, which is sometimes going to be referred to as fat infiltration. We came up with a method to quantify the amount of stir in the muscle. So it's always been one of these, you look at it and you rate it, but this is actually a method now with Dennis Shaw's, a lot of Dennis Shaw's as a radiologist work to calibrate like what you're seeing versus what the AI can do. And then you can make a lot of these other like very sophisticated pixel-based plots that Lara is going to talk about in the next talk. So what I want to discuss today is a study we did, was at our second Wellstone, where we enrolled FSHD patients and they got a biopsy in both TAs at baseline, so the muscles that point your toes. And then they had MRI over time at baseline at 12 months and 24 months. And some of these folks are coming back now at long outcome in our current Wellstone. These are the sort of variables that we're used to collecting. And so in MRI, we're getting fat fraction, we're getting stir. We also did some UTE, which Glenn will talk a little bit more about in the DMD talk to kind of see if we could measure fibrosis. In RNA-seq, there are things like DUX4, IgG and complement, immune markers, extracellular matrix. There was also pathology done, and then the typical kind of clinical data. I want to touch on this for a moment. So I think we've all been really focused on fat fraction, right? That's the thing that we've all heard over and over again. And what we started to see when we could all of a sudden make pixel maps is that the pattern is really kind of striking when you look at how fat is laid down. And so here are two calves, and you can see that they're changing over 1 year. Typically, we would just refer to this as a numerical change. It goes up 0.05. Isn't that cool? But there's a lot of other pattern measures you can imagine. And so if you collapse this down from 3 dimensions to 2 dimensions, you could make a skewness plot where you see that it's skewing to the right more over time, or it's flattening with kurtosis, or the spatial autocorrelation with something like Moran's is changing. And so this is a lot like Doris's plot, but in a visual representation, like you make this plot and it's like, we did it. Well, it's really hard to know what this means, right? So this is RNA data, this is MRI data, this is like all the data we can collect from these subjects. This is just a correlation plot, but it's really hard to make sense of. And so fortunately, there are these tools now, machine learning, AI, these other ways to actually ingest a whole lot of data. And they have some cool features, which is, it doesn't matter how many variables you put in. So you can add all these variables in to try and see if they predict things. Your predictive relationships don't need to be linear, so you can have nonlinear things. And then you can stack data up. And so in our design here, instead of looking at baseline 1224, we can just put them on top of one another. And we're really trying to predict that 12-month interval. So this is what you get. looks a little better than the big giant matrix. And I just want to point out that you kind of see that there are like 3 steps to this model, sort of things that are highly predictive, things that are moderately maybe predictive, things that are less predictive. And I'm going to walk you through what the most predictive thing is in the model. Does anyone want to guess what the most predictive thing is in the model? Not. So it's kurtosis, actually, like the shape of the fat deposition. So the sort of this, something about the pattern is sort of what is most predictive of fat change over 1 year. And then as you march along here, IgG, so an immune marker. So this is the amount of fat. So if your muscle's really normal at baseline, it's less likely to change over 1 year, right? Then we have things like the skewness, Moran's, these other pattern parameters. STIR's pretty far down there, and we can talk about why. Then you have things like pathology, fibrosis from the little sample, complement, another immune marker. And it takes a while before you get down to seeing any of the DUX4 markers. And then something like D4Z4 is also pretty far down there. And so why would this be? Well, the pattern measures do seem to be quite related to if something is changing over time. And Silvia and Lara will talk a little bit more about this. It was really interesting, and Stephen will kind of elucidate this, like, why are these immune factors present and what is the significance of those? And I think most interesting to me as kind of a simpleton with how I think about DUX4 is that that was not as relevant in the model when you have these other terms. And I think that's something important to think about is it's not, we just include pattern. Pattern is the most loaded thing in the model with everything else also present. And so our next steps are really, in our first Wellstone study, we biopsied multiple muscles. And so we're going to be integrating that data. And then in the new Move Plus cohort, we have a whole body scan with individual muscles as well. But where this really leads us is to pigs. So this is sort of, I think, a really exciting parallel that we now have more data to try and address. So I'm going to give you a quick overview of the progress to date, which is really on the imaging side of the pigs. And so if you look in the literature or the internet for a pig atlas, pretty much what you find immediately is beautiful brain atlases of these pigs. But the MRI images are pretty simplistic. So they have scanned pigs, but they haven't actually found where all the muscles are. And so this was really the first step, is to try and scan some pigs, create an atlas. So this is what your atlas looks like. I learned something yesterday, or actually this morning, from Silvia that I did not know. Does anyone know where the hamstring muscle is on a pig? Anyone want to guess? Is it in the front or the back or both? Ham, that's good. Where's the ham? On the table. So what Silvia taught me that's good is that pigs' front legs are like arms. They're not like legs, they're arms. And apparently this is a conserved fact that pterodactyl arms are the same as, so thank you, Silvia. That would have been my special fact, but yeah, so that's helpful. So anyway, so now we have this ability to segment these muscles, which is obviously incredibly helpful because we can make plots like this one on the right. So we can start to look at pixel fat infiltration in the pigs, which is very much where we're trying to head. And so I'm not going to present a lot of data from the initial studies, but I'll get you. Do you?
What the genetics of pigs are?
So this, yes, so this is a tamoxifen-induced pig. Yes, thank you. For the pig, the non-human imaging researchers. Thank you. So yeah, so these are tamoxifen-induced. It's basically tamoxifen-induced DUX4 expression. Is that right? Good. Okay, thank you. Okay, yeah, for sure. So, but what we can see is, so these are just 2 example pigs. So you have your tamoxifen group on the top. And so these are 2 different animals, and you can see we're getting some difference of expression in fat. And so, you know, the next experiment that we're super excited about is to do essentially the same study in pigs that we've done in humans, which looks like this. So this is the study that is almost completed, where they're biopsying two regions of the pig, both in the back, Silvia. And so we'll get biopsies across the whole experiment. We'll get MRIs across the experiment. We'll get blood, and they'll be able to do this similar MRI versus RNA versus pathology. And we also have some, I think what's really exciting is we'll have functional data. I'll show you the next slide. There's some treadmill data and some strength data that we'll be able to integrate as well. I do also think we may need to start calling this whole pig MRI instead of whole body MRI. There's always this WB MRI, so we can work on that ourselves. So this is kind of what the tasks will do to get strength and walking measures. And lastly, I think to test some drugs. I think there's a lot of different compounds that exist and new compounds that you can start to think if complement and these different sort of immune markers are relevant, and we can start to try and knock down DUX4 in these animals in different ways. So I think that's what's really exciting, is once we've done the next study, we'll have the sort of preliminary data. And so just as a summary, I think this fat pattern and immune markers are super interesting, and Lara and Silvia will tell you a lot more about these patterns, kind of predicting progression. The pig data will obviously allow these parallel sort of explorations. And then Stephen will kind of talk about like B cell, T cells, like why is that potentially super useful. And these are all my peeps. So thanks to all the Wellstone, many people were involved in the Wellstone. Sang Song in Missouri did all the machine learning models for this data, which was super appreciated. Folks at Springbok, Doris, my wing person at Candy Krieger, and obviously to all the patients and families that have participated. So thank you very much. Yeah.
Confused about tamoxifen inducing DUX4. That's an estrogen? What's the relationship of tamoxifen and DUX4?
Yeah, this is maybe what get us Stephen. You want to take this one, Stephen?
Oh, sorry.
The question is the relationship between tamoxifen and DUX4.
This is a pig model made by Peter and Takako Jones. It's very similar to their flex mouse model, where the transgene that's put into the pig does not express DUX4 until it's activated by treatment with tamoxifen that creates a genetic rearrangement that creates that constitutive expression of the DUX4. So if you're familiar with the flex mouse, it's a flex pig.
Tamoxifen is just the switch.
So you go through the switch and you can turn it on.
Yeah, so it's basically they have these things where they have switches. Yeah, it's not my area, that's why I understand it.
That's why I punted the question, Dennis.
Wait for DUX4 to turn on. We can turn it on so that the drug assay is a little bit easier to run.
And the big thing with the pigs is that they express, like, mice are very lean, so the mouse model doesn't show as much, right?
Mhm.
And the big thing with that, because we went and talked to Peter, and the mouse model is very lean because mice are just not fatty animals. So the pig is huge because it'll get a lot more,
Mhm.
I don't know, relatable expression, I guess.
Mhm.
Do you want to give the audience all a quick summary?
Yeah, so the question was first, so they didn't hear any. Okay, so the first question, sorry for those of you online, we're just hanging out here, chilling. The first question was the function of tamoxifen in the pig model, and the basic summary being tamoxifen is the switch that allows for DUX4 expression. And the second question related to why the pig, and certainly the pig phenotypically is a better model where we may be able to measure similar things like fatty infiltration and different patterns and those sort of things. So, and I know there's more from hearing from Stephen yesterday. There's many more reasons that the pig is a better model, but I'll let him summarize that. They're cute. They're real cute. Yeah.
Ready for the next one.
Okay, great. Okay, so we're gonna get ready for Lara's talk now.
I'm Lara Riem. I'm the Director of AI and Data Science for Springbok Analytics. Thank you so much for having me. I think this presentation fits really nicely in with the last series we just had. I'm really just mainly going to go over methods for calculating fat distribution. And then Silvia's presentation next will show how powerful those metrics can be when you look at it across multiple patients. So first we start with MRI. This could be the full body, it could be a certain section. And we put it through our AI to segment the individual muscles and bones. So here's just an example view of it from a slice, and then here it is in 3D, so you can kind of get the visual of the different regions. And then next we just double check the AI output and vet it to make sure it's correct, and we get that final 3D model. So right here, I just wanted to show some of those inner muscles that you can't see necessarily, and then the outer muscles here. So one point of discussion too in many fields is looking at the muscles grouped together as a function or as individual muscles. We look at individual muscles mainly because of two reasons we found. And I can use this quad here as an example. So in this patient, if we were to quantify it as how much fat infiltration is in that whole region, with low being that cream color to high being that orange color, you could see if you kind of group all of them together, you might lose some differences between the muscles in the functional group, because not all muscles within that group might be progressing the same way. So in this example, you could see this muscle is a little bit more progressed than this one, and that's maybe a distinction we want to know. The second reason is if you group all the muscles together, you're also including intramuscular fat, so the fat between muscles, which for a case like this where the muscles are close together might not be as big of a deal. But if the muscle starts to atrophy, you could see then we're considering a lot more intramuscular fat as part of that grouping. We might be overestimating fat infiltration. So the big thing we want to do is make a distinction between intramuscular fat and intramuscular fat, the fat within the muscle. So that's kind of what we're showing here, is we could segment the bone muscle boundary and then just look at how much of that muscle is being replaced by fat infiltration. So then using those two things, we can separate it, right? We can look at muscle size and then the fat infiltration separately. So this is an example of 8 patients with FSHD. We wanted to show the differences in muscle development pattern between patients. So we know that an expected muscle size based on a patient's height and weight, and we can highlight that with red and the orange being a smaller muscle than we expect, this gray being normal, and then blue being a higher or larger muscle than we expect. So this is important because these red muscles could indicate, oh, there's atrophy, maybe the muscle's not being used, or there's active progression. While hypertrophied or larger muscles might indicate, they're getting larger because of increased fat infiltration, or maybe they're being used to compensate for muscles that are being progressed. So looking at the other side of it, right, looking at fat infiltration of those same muscles, we have the same patients, and we can look at overall fat infiltration, once again, low to high. So, and this has been discussed in the previous presentations, but the important thing is, fat infiltration occurs in different muscles for everybody, and the level in which it occurs is different for everybody. So the main purpose now of this presentation is diving into that. How do we look now inside each muscle? So in the MRI, we get the water phase and the fat phase, and we can then pixelate your muscle, right? We have it in 3D. We can quantify then for every pixel in that muscle in 3D, how much of it is fat and how much is water or muscle. So the more blue, the more contractile muscle we would say that pixel is, while this purple to red is kind of in between, maybe transitioning, while the yellow indicates more fat. So here are 3 example patients. This is their soleus calf muscle. So this patient is non-impacted right now, so all blue. Well, this patient's starting to have some small progression, mainly at the bottom of their muscle. And then this patient has higher progression. You see more fat than muscle. You also notice that the shape starts to change, which is also something that we can characterize with that muscle size. As Seth discussed, there's 3 ways we can then actually like take this visual representation, right, and actually quantify it into numbers. So using that same example, fat variation, it's how much, the fat varies throughout the muscle. So really, how many different pixel colors are we getting? So you can imagine that if someone's actively progressing, like they're having active fat infiltration, their variation is going to get higher, right? We're going to see different pixelation colors. Well, on the other side, as Seth was showing, if you kind of put this into a 2D graph, so if you looked and counted, how many times do we see 0% contractile muscle? How many times do we see 100, 80, 90? We can kind of stack that up and count how many times they each occur. So just visually looking at this, you can tell that we have more of the fat-infiltrated pixels showing up than the blue contractile, and that's what that histogram shows. So skewness tells us how much the pixels are lining up to the fat-infiltrated or contractile muscle. So if it's negative for this patient, it was negative 1.3, indicates it's more skewed to fat infiltration. Kurtosis tells us kind of another way of how much variation there is. So you can imagine that in that previous example with all the contractile muscle pixels, they'd probably all be stacked right here, and you have very, very, very little variation. So your kurtosis values can be high. Well, in this example, it's more spread out. You have more different pixels, so you have 0.52. Lastly, you can look at the clustering of the fat infiltration relative to the muscle. So for an example, in this case, you can see that clearly the contractile muscle is clustered in one area, while the fat infiltration is clustered in another, which looks kind of like a black and white depiction, which gets a value of 1. On the flip side, if it was more like checkerboard, where you have the fat and then the contractile muscle kind of alternating, you'd have a value of -1. So this is just a way to see how clustered that fat is. So other than a single value, we can also look at it as a distribution. So looking at it slice by slice, we can see, okay, how much of it is fat, how much is muscle. For this example, let's say you're looking at me straight on. If you're to look at the muscle from like my feet moving up to my head, so from 0% to 100% of the muscle, we can see that there's more fat on the bottom. So increased fat fraction or fat infiltration that slowly goes down as we start seeing more contractile muscle. So starting higher with fat and then lower as fat, it becomes more muscle. So focusing on how we can quantify this, we found there's 5 different patterns. So you get that pattern where it starts with fat infiltration on the bottom and then more contractile on the top. You see vice versa, where it's more fat on the top with muscle on the bottom. You also see kind of alternating where there's fat on the ends, but nothing in the middle, and vice versa. Fat in the middle and contractile muscle at the ends. And I'm going to show you an example of each. So here's an example of vastus medialis on quad muscle, in which you could see there's a lot of fat on the bottom and then contractile muscle on the top. So this distribution, you have increased fat from 0 to 40, and then it starts to go back to muscle. We would expect if progression increases, this graph's just going to start shifting to the right. With the deltoid here, we see the reverse, contractile muscle at the bottom with fat infiltration on the top. So once again, low to high. Here's an example of the ab muscle in which there's fat infiltration in the middle and then contractile muscle at the ends. So you see that low fat infiltration, high, and then low. For this one, we could see that fat infiltration start progressing on both ends, where it starts to move this way and this way. In the reverse, in this quad muscle, we see that it's fat infiltration on the bottom and then contractile muscle in the middle. And then for this one, we would see probably progression creeping inward. Lastly, we also have a case where the fat infiltration can just be constant throughout. So you can see with this calf muscle tibialis anterior, it's just kind of consistent fat infiltration level. So one thing we can do, and we did this for about 17 patients, is we can say, okay, for this muscle, your distribution pattern was one of these 5. And we can look at this across all the muscles and across all 17 of those patients, just to see, is there a specific pattern? Do some muscles always have a fat infiltration that progresses this way, or there's always a pattern this way? So here's some just example muscles. So just pointing out the soleus here and the tibialis anterior. You could see they're more likely to have that near to far, so where the fat starts on the bottom and starts to move upward, distribution. But they do have a occurrence of other distribution patterns. But it's good to know, like, some muscles might be more likely to have progression in one way versus another. So tying this all together, why is this important? Why should we care about distribution? So in this example, this person's gracilis muscles right here, you see one on the left side, it's actually progressing from top to bottom. On the right side, it's bottom to top. And after a year, their total fat infiltration in the muscle only increased 1%. However, if we look at the distribution, we see that it's doing that movement progression forward. And it's actually quite a bit. And this is important because, you know, this person could still have really good function with their gracilis. But that's just small 1% increase, suddenly they could lose function in that muscle. And it's mainly because where this progression occurs can greatly affect the function of the muscle. So this could suddenly disrupt their ability to use it. And so that's why it's important to know where this progression is occurring on top of how much there is. And I think Silvia will be touching on that in a lot more detail. So I'd like to thank our research collaborators and our funding. We couldn't do any of this without that. And I'll take any questions. Let's see here. How is the angle of the 2D flattening from 3D? So it's not like we're technically, it depends on which way you're saying flattening. When we're looking at and doing that histogram, luckily the angle in which we're capturing doesn't matter. But as far as the distribution, we do just assume for now that the patient's legs aren't angled in a way that if we're looking from inferior to superior, that's greatly influencing how like the slices are taken. And we've done a few analyses to check that assumption. But there are ways we could in the future register it so the muscle is always very like rigid, making sure that any possible change in patient placement of their muscles could impact that value.
With the exception of the tibialis anterior example, which showed diffuse fatty infiltration, all the other patterns show contiguous spread. And that suggests that there may be like a trigger and then some intermediate toxicity or factor. Are you thinking that this somehow informs the biology of disease spread? And what are your thoughts about that?
Could you repeat it? I heard a little bit of it.
So I think the basic question was, you know, how these patterns might inform kind of that early stage of the disease to, you know, the actual muscle being affected. Is that, or was there something about this? Hold on one second, Lara, we're going to reinterpret.
It seems that most of the spread is contiguous, as if there were a contiguous, as if we were a factor that's promoting toxicity. It starts in one region and then it's kind of
Creeping from that.
Is there an idea behind the biology of what this implies, or?
So I can't speak to that necessarily. I'm not as familiar with how that might, like the mechanism behind that, but I agree, like based off of what we're seeing with even though the distribution patterns are different, it does seem to be like a rule that once it starts to move out from that area, right? It seems very rarely we see it kind of like pop up in different areas and then spread from there. it seems like it's gotta be, it comes from one, or maybe if it's the two different ends, but most likely it seems to be coming from the ends of the muscle.
So again, I'll approach it as something that I don't have the expertise in, but I know people that do. Not them. So again, I'll approach something I don't have the expertise in, but I know people that do. Yeah, no. But as far as the idea, my understanding is seeing DUX4 is a relatively rare thing. So the idea that there might be a wave is really would fit with it. Daniel, for instance, you could make one. Like I said, I don't see Steve, right? Oh, there he is. Here.
No, I think it's a good question that was asked, and something that, that we're actively trying to answer on a molecular level in terms of what the spread through the muscle means. We have a number of mechanisms right now that suggests ways of spreading the activity along the muscle, and then I talk about hypotheses. The simple answer is we don't really know, and we're trying to find out.
Thank you. Okay. Thank you so much, Lara.
Yeah, I can answer the other two at the panel discussion if you want.
Okay, sounds great. So the one thing I just want to say about Silvia, which is her savant skill, is that anytime you have a muscle question, I feel like I've been so focused in the field over the last 15, almost 20 years now on fat fraction. And every time I talk to Silvia, I learned something more about the kind of questions that were raised, which is these muscles are working, they're doing things. And I think that we oftentimes are very focused on these cellular mechanisms and other things, but the injured muscle's working and trying to compensate and trying to do what muscles do. And so I think Silvia will be a wealth of knowledge in the way she's thinking about this stuff, which is more like put everything back together again. So.
Well, thank you, Seth, for that introduction, and thank you for the opportunity to speak here. It is exciting to be able to chat with you about the progress that we've made, really thanks to Friends of FSH and the funding and support. So as Seth said, I'm at heart, I've been studying muscle physiology and mechanics for my entire career. I'm two things. I'm a biomedical engineering professor, and I also co-founded and started Springbok Analytics. It actually came out of research that I was doing in my lab, all developing new techniques to quantify muscle for a range of applications. Actually, the first application that we were interested in was understanding and being able to characterize muscles in children with cerebral palsy, and we've sort of blossomed. We look at athletes, we look at lots of different populations, but I've had a longstanding interest in neuromuscular disease and neuromuscular disorders. So I was really excited when Seth reached out and said he had this data set. Because actually, the other interesting thing, believe it or not, that most of my career is about how muscles are heterogeneous and how that happens and why that happens. It's literally the purpose of my PhD dissertation. So when you were showing me that data, I was like, oh, we got to check that out and really try to figure it out because it's complicated and an important problem. So with that, I'm going to start. I want to, before I talk about putting things all together, I wanted to take a few steps back and kind of put into context where the MRI really fits in our kind of understanding of FSHD. So, the discovery of DUX4 and that as the mechanism of FSHD obviously was a huge breakthrough in understanding the disease, right? Like now we understand where the target should go and what the underlying biology of the disease is. It's a huge step forward. And so that's really, I think about when we think about muscle at the kind of and muscle movement, it's really what I call a multi-scale process. So at the very small scale, you have the cellular molecular mechanisms, right? But if you want to think about how muscles actually work and how that affects the way that you can move, there's a lot of things in between that influence that, right? So next step up is now you put all those things together and you think about, okay, you have lots of cells in your muscle, and they've kind of combined to form a tissue, and they have their own properties and architecture and everything. And then you got to zoom even further and say, okay, within a muscle, you have a whole bunch of that. And there's a way a muscle is put together that influences the way it works, right? That's something that we call muscle architecture, but that influences how it works. And then if you go to the next level, you think, okay, well, muscles work together in groups. So not only is the way the muscle is put together is important, it's how they work together in a group and how they're arranged that influences how they work. And then you put that all together into a whole body, and how all of those groups are organized together and work together influences how you move, right? So, you know, when I think about how human movement works, I look at it this way because it's a really important thing to do when you're trying to understand, especially complex diseases like FSHD, which really it's this multi-scale kind of problem. It's a very small scale. If you think about scales, I'm an engineer too. So we think about like scales and numbers, like this is all happening at the, you know, at the nanometer scale and piconewtons, we say, but really what we care about things are in the meter scale and in newtons or thousands of newtons. Like that's what matters in terms of the actual function that our body needs to do. So, so that's, but interestingly, in FSHD, you know, really what we're trying to figure out is the relationship between this scale and that large scale. But how do you connect those two, right? There's a whole bunch of stuff in between, and it's super complicated. We just looked, that was like very simple diagrams illustrating those things. And they all are affected by FSHD too. Oh, did something just happen? Hello? Can you guys? Oh, sure. Hello? Ah, that's better. Oh, better, cheddar. Okay. There we go. Is that okay? I'll just hold it. I'll just hold it. Oh, here. Okay, that's easier for you. Thank you. Sorry. So, you know, we were learning about these things, right? At the tissue scale, we have this heterogeneous fat infiltration and fibers are affected. At the whole muscle level, you have overall muscle atrophy and changes at that level. And then even within groups, we're realizing that within a group, you have some muscles that are affected and others not. And all of that leads to the, you know, complex functional impairment. So, but right now, if you think about clinical trials, you know, we'll even, you know, talk about the Fulcrum trial. They had a treatment down here, and their measurement of how it worked was all the way over there. So essentially ignoring all the parts in between and expecting that you're going to get this magical relationship that just comes out, right? You know, the chances of that are going to be pretty low, right? Especially as you're developing treatments. Maybe once there's an absolute cure, it'll be much more obvious. But until then, if we really want to make progress in treatments, you have to think about the in-between. And that's really where the MRI comes in. Kind of the whole point is like the MRI helps us understand the connection between these two. We can look at how now we can measure at this sort of like, I call it regional or tissue-ish level, like what's going on. Those details about what Seth and Lara were nicely talking about describes the, you know, the pattern of fat in progression at that tissue scale. Then we can move up at the individual muscle scale and see how things are affected at that scale in terms of fat and lean volume. And then we can even look at the implications at the group level. And ultimately, with that information, it can actually then inform why or why not you might be seeing a change at the functional level, right? So it's really the idea is connecting the dots. So that's the first concept I wanted to just bring up, is the idea that like that's where these MRI measures really fit in, is connecting the dots. But the other thing that you may or may not have been thinking about was, well, that's a lot of information. You know, it's a little simpler to say like, okay, I'm going to treat at this scale. I'm going to measure something simple like reachable workspace. And it's very obvious how to design your trial, right? There's just like it's much simpler to do a power analysis and all those things. So how do you handle all this detailed complexity? It sounds great. So that's the challenge that we've been working on in the last year, thanks to the money from Friends of FSH. So just sort of wrap our head around the challenge. So you think about this, okay, now we're adding all these new variables that you want to consider in quantifying and understanding a disease. Now we're thinking about, let's say, okay, we're going to do this regional thing. Let's say we define 8 regions within each muscle. Let's say that we're going to split it up into 8 regions so we can look regionally. And then we saw all these really interesting metrics like kurtosis, skewness. So let's say there's 5 metrics per region. Let's say we have that. And then we have 4 muscles or more per group, and at least 2 metrics per at the whole muscle level. And then we have, if you count right and left, we have 32 or more primary muscle groups in total, and maybe 2 metrics per group. So how are you going to handle that? That's if you kind of count it up, over 20,000 data points per patient. So that's a lot of information. So what do you do? And then even there, then of course you have clinical trials with over 100 patients. And, that's actually a lot, but also maybe not enough if you really want to look at all these variables in a pretty more standard way. So how do we address this problem? Well, here's what we're going to do. Our idea then is to integrate all this information to create, I'm using kind of a buzzword here, digital twin. You kind of hear this term here and there, but that's kind of what we're after actually, is essentially a digital twin of each patient that incorporates all these details into one kind of singular computer model. So essentially taking and whatever information we can get at the molecular level. Seth was talking about some nice example how you would integrate things like DUX4 measurements or IgG, things like that, maybe from a biopsy, but other metrics. But then we have all these other metrics that then we can integrate into this machine learning based model. We use specifically random forest method. And then with that essentially digital twin, we can then identify that person's predicted personalized progression. Essentially, how will that person change? Which muscles will change or should change based on this digital twin? And so if you know that for a particular person, then you can say, well, how does that compare to what actually changed? So if it's the same, then maybe the treatment didn't really change much. If they've progressed less than predicted, then that means probably the drug is doing something, or vice versa. So that's the overall vision, is a way to really address this complexity that we have, that we know this heterogeneity in the disease that we know is really hindering our ability to see if a drug works in this kind of digital twin way. So a little snapshot into how that works. I don't want to go too much into the weeds, but I also wanted to show some data. So this is how generally it works. And Seth gave a nice introduction to this type of modeling. But so essentially what we do is we call each data point actually either a region or a muscle, and we can incorporate many different metrics and ultimately to predict another metric. And so the way this is kind of a sampling of the data set that we've been working with, and it's this data lake, and it's really powerful because each study is somewhat limited. Doris described nicely the 30-person study that data is very methodical and beautifully done, but she talked about how much work it was just to get 30 patients. So she's put that data set into this data lake, and we've gotten more data from the Wellstone Center. We have some data from a project with FSHD Global. We've put in some Move Plus patients, and Fulcrum has shared some of their phase 2 placebo data that we've put into there as well. So we're kind of amassing data. And so this kind of gives you a sense of what type of, and so here's a little bit about the model. Hopefully it's not too technical, but so the way we've put together the model is sort of inspired by this multi-scale concept that I was just talking about. Because truly, machine learning is helpful, but you don't want it to be designed in a way that you have no idea what's inside of it, right? Because then it's really not insightful and you're not actually really sure what it means. So we've put it into stages. So there's a first stage that focuses in on this sub-region. Essentially, takes each muscle, breaks it up into regions, and assesses our ability to predict change at that level. So change in the fat in the fat and within that level. Then the output of that model is fed into another model that now is at the whole muscle level. So it takes that information, like what is thought to be changing at the regional, along with other information. Some we have like D4, Z4 and things like that incorporated there. And then that then is used to combine, essentially calculate what would be the associated group level to then ultimately predict a change in function. So it's sort of aligned with how movement works, essentially. And so the way that we parse out our data, this is an important part of the process, is had I described the whole data set. So we took 10% of it to train the stage 1 model. Then we took 70% of it essentially to, in parallel, train the 2 and 3 models. And then we reserved another 20% that's totally naive to all the training to then test out the predictions of the model. So just a little snapshot of its performance. I just chose, there's a lot of stages and everything, but this one's, I think, a nice one, and one I wanted to bring up, that lean muscle volume change is very important for function because indeed, like, it's the muscle that's driving function. But so these are results from the left is what we get once we've trained the model with the training data set, see a very nice correlation. And then the associated testing data set performs quite well. Errors in the mean error is almost 0, and the root mean square error is about 8 milliliters, which is pretty small relative to a muscle. So we can pretty well predict the change in lean volume, annualized, I'll say, in each data point is a muscle. And the other thing that we get out of it, which is essentially, this is the type of plot that Seth was showing, but kind of visualized a little differently, is how each parameter influences the model prediction, essentially. And so I just wanted to show this briefly. Things like D4, Z4 length are in there. This is the output of the regional model. And actually, it turns out that bone properties also influence the outcome. I was just talking about, I forget who we were just talking about bone composition. And it turns out that some of those parameters actually influence how muscles are they're tracking with how muscles are changing, which is an important thing. And then the other thing, I wanted to show you a snapshot into that last stage of predicting change in function. And here we had timed up and go. Unfortunately, with this data set, since we're sort of piecing it together, it was hard to find functional measurements that were done in the same way and longitudinally. So this sort of narrowed down the possible data, but we still were able to predict timed up and go changes from these group level changes, showing which muscle groups influence the outcome. And of course, the initial, the timed up and go at the initial time point also influenced how it was changing. Obviously, if it's very fast, then you're not going to change a lot. And if you're, at a certain region, it's going to have an impact. So this is giving you a sense of our ability, kind of the concept of then ultimately going to stitching it together to predicting how a particular patient may change the function. So just back to the big picture, we're excited about this. We're about to submit a paper to that gives this preliminary model framework and the results we're getting from this data set. But there's a lot to kind of look forward to that we really think it'll be exciting to do. One is adding more data types. We talked about STIR. I think that's a natural one that we'll add. There's a lot of talk about blood biomarkers. So this like circulating factors, obviously that could play a big role here, and that would be a really interesting thing to incorporate. Other biologic factors. Obviously, more data points in patients is useful, especially in that has more function with more functional data. And then also incorporating treatment will be another really interesting thing, essentially, potentially as when you're looking at a trial like potentially the phase 3 data, which Fulcrum has agreed to share with us, if we add treatment as a parameter, does that show to influence outcome, which could tell you if that treatment worked or, had an influence. So that's the overall picture. And with that, I would again like to acknowledge my great colleagues here and funders and you all for your time. Thank you.
So Sylvia, we do have one question from the chat. Can you help us understand how we take this detailed information to a clinically relevant endpoint that's adequate for approval?
Yeah, so the idea is kind of what I was describing, where with this, essentially, you could think about it as a digital placebo. And so that placebo is going to tell you what the predicted change in whatever the endpoint variables or variables that you've decided to select, likely functional metrics, because we're showing that we can take the model to that point, especially once we add more data. And then you would say, you compare your treatment data or the person on treatment with what the model predicted it would be. Essentially, so in some ways you can think about it as a very tailored placebo for that person.
Okay, thank you. So we're going to make a little bit of a transition. We've given you our best shot of all the MRI complexity we have. And in this half, we're really going to kind of take a little bit of a sidestep to both a lot of the molecular sort of mechanisms that Stephen Tapscott for decades has been really a pioneer in this field of understanding FSHD and trying to integrate some of the MRI features that we're seeing with the molecular features. And then we're going to, we'll have Leo talk a little bit about non-MRI biomarkers, because this is still a huge topic in the field, or what are the other types of things that you can measure with DEXA or with some other sort of more muscular conductivity sort of things. And then we'll hear from Glenn Walter and Krista Vandenborne in Florida, who've really pioneered DMD, sort of the functional study that was the long-term follow-up of DMD boys that's led to a lot of really amazing trial implementation. And then at the end of this session, Jeff will kind of bring us home with trying to frame all these things in terms of what does this mean for biomarkers and trials in FSHD. And then we'll have a panel discussion where hopefully we'll get to a lot of the sort of top level questions. And so the only thing secret I can say about Stephen is that I'll have to out the question, because otherwise he'll bring it up at some point, which is Stephen for years has been asking me, well, what about the lymph system? And so, I'm sure that if it doesn't come up in this session, in future years, we'll no doubt discuss the lymph system. So thank you, Stephen.
Okay, thank you, Seth, and thank you, friends, for the opportunity to talk to you today. And thanks to members of my lab for coming as well. And hopefully you'll be able to ask them questions afterwards if there are any later. I was going to focus my talk on the molecular correlates of some of the MRI changes.
Okay, great.
Okay, as Seth was, as some of the other talks, I think in particularly Seth's, he attributed many things that I hadn't planned on covering to my talk today, but we'll see. We see how it goes. And how do you move the slide forward?
I think I've booby-trapped you here. Okay, now you got control.
Okay, and so the disclosure slide, I've consulted for a number of companies. So what does the MRI tell us about FSHD? And that was really the topic of the Seattle Wellstone centers. That in the first Seattle Wellstone, we did a longitudinal study of doing muscle biopsies and looking at the MRI correlates of the molecular studies for RNA sequencing and pathology in those studies. And then the second Wellstone study was doing bilateral anterior TIBs biopsies. And in all of those, we had histology and RNA sequencing and functional measurements that we could correlate with the MRIs. And there have been several papers published focusing on this, and additional papers from the group describing the work that's been done there. And I'll just summarize a little bit about what we've learned. And so if you look in the RNA sequencing and get a DUX4 score. So genes that are regulated by DUX4, the higher the DUX4 score, the more DUX4 is expressed in those muscles. And you compare the DUX4 score from control subjects with biopsies of control subjects that don't have FSHD to the muscles that were STIR positive when they were biopsied in an FSHD subject. You can see that there's a much higher DUX4 score in the FSHD muscle. In fact, the score in the control muscle is virtually 0. But what's interesting is in the normal muscles, the ones that have a normal MRI characteristic, relatively normal pathology, you also have an elevated DUX4 score compared to the unaffected muscle. And so this has led to the model that over the years you have low levels of DUX4 and its target gene expression for probably many years in this muscle. And then as that amplifies over time, it starts changing the pathology of the muscle and starts changing the MRI characteristics of the muscle. And one of the nice things about RNA sequencing is you really look at all of the genes expressed in the muscle biopsy. And so you can look at baskets of genes that represent inflammation. It's been known for a long time that there's focal inflammatory changes with T-cell infiltrates in FSHD muscle. And you can see that those increase really in a way that correlates with the DUX4 score. ECM or extracellular matrix genes increase over time as well. One of the ones that is interesting to us is a basket of genes in the complement pathway. The complement pathway is one of the most ancient immune pathways that protects against invasion of foreign species. And so, for example, if a cell is contacted with a foreign bacteria, it will activate the complement pathway through the innate immune signaling system. And the complement pathway has a cascade of events that eventually forms this complex called the membrane attack complex that it secretes into the surrounding intercellular space. And that membrane attack complex essentially punches a hole in the bacteria and kills it, right? And so it's interesting that the complement system is activated in FSHD, and we can actually use antibodies that recognize this membrane attack complex and show that that's present in the skeletal muscles in FSHD. And it's one of the earlier findings. And it's one thing that we're thinking about, although we don't have any evidence to support this, is it really making functional membrane attack complexes so that one muscle fiber that's activated the complement system might actually be punching a hole in the neighboring muscle fiber? Is this a way that the effect spreads between muscle fibers as the entire muscle gets affected by the changes in the initiated by the initial DUX4 expression in one subset of nuclei? Another thing the complement system does is interact with B cells. And so it stimulates B cells to make their antibodies. And, again, you can imagine if a bacteria is there and it's being stimulated, the complement system will help the B cells make antibodies to the bacteria. And in that regard, it's interesting that immunoglobulin genes are also highly expressed in these STIR positive muscles where you see the high DUX4. And the muscle doesn't make the immunoglobulin genes. It must mean that B cells are infiltrating the muscle in order to make those immunoglobulin genes. And so maybe there's some immune component, not only from the T cells, but also from the B cells that's helping the muscle progression. So besides the biology, we can use these baskets of genes to, hopefully look at response to therapies, for example. What you'd like to see is if you had a STIR positive muscle and a high DUX4 score expression, if you treated with a drug, you'd lower that maybe to the earlier level. But it also means you can look at muscles that are not STIR positive and see if you push that down to the unaffected subject level as well. And so we're hoping to use these baskets to do this. And this is one of the things that Avidity has incorporated into its clinical studies. One of the things that surprised me, though, about the bilateral study was that if you looked at the left and right tibialis anterior from the same individual, if they had a high fat infiltration in their, for example, right anterior tib, they would also have a high fat infiltration in their left anterior tib. And you could possibly explain that by saying, well, they're just more advanced in their disease process. But it also raised the question of, is there a communication between one side of your body and another side of your body, or between diseased muscle in one part of your body and the other? And that was also kind of brought up when we looked at the molecular signature in the right versus the left. And so if your DUX4 score was high in your right tibialis anterior, it would also be high in your left tibialis anterior. And that was irrespective of whether you had changes in your MRI characteristics in those muscles. Complement went in the same correlation left to right, and particularly the immunoglobulin genes. Again, these are coming from infiltrating B cells. Now, infiltrating B cells also go in the circulation. And so there you can actually have a really easy to understand model where if maybe you have some B cell stimulation in one muscle, it gets into the circulation, goes to the other muscle, and amplifies it in that muscle as well. And so that's really something that we're thinking about now. Why would you have a B cell stimulation? Well, DUX4 is normally only expressed in the early embryo. It activates genes and proteins that are made only in the early embryo. And the early embryo hasn't really established its sense of self from the immune system. And so if you re-express these, they should be immunogenic. And so maybe the B cells are responding to some of these DUX4-induced proteins, or perhaps even to the DUX4 protein itself. I'll get back to that point in just a minute. And so what does this right-left correlation mean? It suggests that there might be a whole body progression. We think about muscle by muscle effect of FSHD, but superimposed on that, there's the whole body progression in FSHD. And over time, all muscles will progress kind of at the same level, at least on the right-left of the same muscle group. And so that brings up the hypothesis that I've been alluding to, that progression of one muscle might increase the progression of other muscles. And so how would that occur? And it could be from an immune response. I mentioned the stimulation of B cells, perhaps by a DUX4-induced antigen. People have looked for antibodies in FSHD to the muscle in FSHD, and they haven't found them. And so that simple hypothesis doesn't have any substantive scientific basis yet. With funds from the Friends of FSH, we're doing some single-cell sequencing on cells derived from muscle biopsies to try to find, what are the sequences of the T cell receptors and the B cell receptors, and can we get any direct evidence that there might be an immune stimulation of specific T and B cell populations in the FSHD muscle that would support this hypothesis. And another possibility is maybe there's a secreted factor from damaged skeletal muscles, cytokines or microRNAs or some DUX4-induced protein that you would normally think about these as potential circulating biomarkers, but you have a circulating biomarker. That biomarker itself may have activity that could affect disease progression. And so what do we know about biomarkers in the blood? Again, the biomarker in the blood would be really good because it would measure whole body disease burden. If you do a muscle biopsy, you're presumably just measuring one spot in one muscle, and it'd be great to get a whole body disease burden measurement. MRI can integrate over many muscles, but it might not show the rapid change that you would want in a biomarker that would show, for example, a response to therapy. And so what are candidates for one of these circulating biomarkers? Again, what people have looked for, including our lab, are proteins or genes induced by DUX4 that would make proteins that would be secreted and could be found in the blood. And so as one example, placental alkaline phosphatase, which is a DUX4-induced gene that makes a secreted protein, and it can be found in the tissue culture supernatant of DUX4 expressing cells. And in a paper by Suja Jagannathan, they looked in plasma, but the problem was that there was already a high enough level of this protein in the plasma. They couldn't distinguish subjects from FSHD subjects from controls, but that's the principle that we're looking for. Avidity has reported that they are looking at a DUX4 induced protein that they can identify in the plasma. They haven't disclosed the identity of the protein, but again, it's proof of concept that there could be other proteins as well. And so what could they be? They could be inflammatory genes. The Jones group and others have looked at IL-6, TNF. Alpha has been identified as a gene induced in the FSHD muscle that could be detected in the plasma. So there could be these inflammatory components. People have looked at secreted microRNAs. And then I mentioned complement before. And we actually looked at complement in FSHD versus control subjects. And so, for example, complement C3 and C4b are really elevated in the plasma of FSHD subjects compared to control subjects in a statistically meaningful way. Clearly there's a big spread, and you can't use it to, say, diagnose an FSHD individual versus a control individual, but it really suggests that circulating features such as the complement that's activated in one muscle in FSHD could affect other muscles in FSHD. And so you'd really like to have a model where you can test some of these things. I mean, there's not a justification in humans at this point to knock out your B cells or knock out your T cells, because you would have to do these long-term clinical trials. And so one of the models that I'm very excited by is a model that's being developed by Peter and Takako Jones together with Exemplar Genetics, the pig model that you heard about earlier. And so why is the pig model really so attractive? Well, if you express human DUX4 in pig muscle cells, you really activate virtually the same transcriptional program as when you express human DUX4 in human cells. And so these transgenic pig models that have an inducible human DUX4 really have a chance of activating the same pathways. Now, that's not the same in mice. Human DUX4 and mouse DUX have evolutionarily diverged from each other, so they have slightly different binding sites. And while mouse DUX and mouse activates the same genes that human DUX4 does in humans, if you take human DUX4 and put it in mice, it really does not do a very good job of activating that DUX4 program. But if you put it in pigs or dogs or many other species, it really activates the program. Rodents are the outlier. They've diverged away, making them a particularly bad model, or not. They're a good model for certain aspects of FSHD, but if you want to look at some of these downstream systemic aspects, some other species such as pig are better. And so again, the DUX4 in pig really recapitulates the gene expression. And as was mentioned earlier, inducible pig has been made by the Jones group, and currently there are functional MRI and molecular studies in progress. And if this does support the hope of a good model, you could really use this to look at the role of the immune system in disease progression by modulating T cells or B cells or other systemic aspects of FSHD. And you could really sample lots of tissues as well that you can't do in humans. And so I've told you about what we've been doing in the past with the past Wellstone studies in the Seattle area. And I just want to mention that we're really trying to put all of this information together in the current Wellstone funding period to do an interventional study with clenbuterol, a drug that came out of Franz Sverdrup's screens and other people's screens as suppressing DUX4. It's a safety study, but we'll also be using it to test the measurements of the MRI and molecular features to see how well they report on the activity in these studies. I want to thank all the members of my lab, both past and present, and particularly thank friends of FSH, and thank you guys for listening.
Okay, we have time for one question. I have a question. I was thinking that the circulating biomarker or antibody hypothesis kind of requires that then induces DUX4 in the muscle on the other side of the body. Is there any hypothesis for what might happen in between once one muscle is infected and starts to infect another muscle?
Yeah, there's a number of different studies suggesting that cellular stress can de-repress, DUX4 and FSHD muscle, and so there could be simple stress response pathways that are downstream of that.
Leo Wang, my counterpart. Leo has just been an incredible neurology resource for me in all of our Wellstone studies. And I think we're finally getting to where we wanted to get to, which is actually be able to put the MRI, the functional data, and some of this other data back together again. So it's an exciting time. We can finally write all these papers that we've wanted to write. So take it away, Leo.
Thanks, Seth. You let me off easy. I was really worried. And I also see that I only have 16 slides and Dr. Tapscott had 18 slides, and he's definitely more of a professional, can get through a lot of information in a short amount of time. I think that here at University of Washington, we're really just fortunate to be in this region and to be in a place where we get so much support from Friends of FSH Research. So we're really appreciative of the help and the help of the community of patients and the community of caregivers here. And we've been thinking a lot about FSHD and participating in a lot of these national, international consortiums. And I just want to talk a little bit about some of the stuff that we've been thinking about. And when we talk about biomarkers, we're really talking about things that can measure FSHD and measure FSHD changes over time. And we're just going to talk a little bit about some different biomarkers and really continue on and talking about what Silvia was talking about, which is, I thought, a great way to explain it to people in terms of, when we're looking at a trial and we're looking to see how things change, we're looking at things on different levels. We're looking at things on the organism level in terms of how it changes in strength and how it changes in how you move, how you get up, how you work, how you do your daily function. We're looking at things at the muscle level, and we're looking at it in terms of a group of muscle, in terms of individual muscles. And then we're looking at it at the cellular level and looking at the whole muscle itself, which is a muscle cell is basically runs the whole length of the muscle. It's one long muscle, and it's different than a lot of the cells in our body in that there are multiple nuclei in there. So it's really different. It's like one long tube that goes down your whole bone, and there's multiple nuclei there, and each of them have DNA and have a repository of genetic information. And how does that change over time, and how does that change in a disease? And finally, Dr. Tapscott was really talking about DUX4 and how that changes the disease and how that gets turned on. And a lot of those things we don't really understand. And a lot of those things will be, answered with the pig model, which is basically, I think, as a research community, we think is a really good way to think about this disease, because it doesn't really occur in mice. And so having an organism that's closer to humans and that we can manipulate and understand the disease changes, what would be good. So there are two things that I want to talk about. One is the DEXA scan. And so it's measuring muscle with amount of muscle with DEXA. And so what DEXA scan is basically it's basically an X-ray of your body, and based on how much of the X-ray gets taken up by your bone, by your muscle, and by your fat, we can put together a map that gives us a sense of how much muscle you have. And you can divide that, and after the images are done, and looking at it in a limb and looking at it in your torso. And so this is just a really nice test because it's really low energy in terms of X-ray. It's really minimal exposure. It can be done really fast, and the processing is not as extensive as required for what we're doing for MRI. And so we're trying to figure out, how useful this test would be in measuring change, and how useful this is in reflecting what we're trying to measure. So we're trying to measure the amount of muscle. Does that really reflect in the amount of strength that each of that limb is representing? So this was part of a international consortium that Dr. Statlin organized, and was about 185 patients. It's in 8 US sites, and there was also patients from Netherlands and Italy. You can see here that the age of first symptoms with the median age was about 19 and a half, and the age of diagnosis was a few decades, 1.5 decades later at 35, and then the age at which the patients came to our research visit was at 52. And when we looked at the DEXA scan, and DEXA scan of the right arm, and really trying to compare it to the strength that we measured with some of the tools we did, this is what we found. We found that the amount of lean body mass, which is the amount of muscle that you can measure on the DEXA scan of the right arm, so like measuring it from here on out in this DEXA scan, that's on the x-axis. And you can see that the more muscle, which is towards the number 5 end, the more strength you had, which is the y-axis, and then the more strength you can exert with your right arm in terms of using your biceps, using your triceps, raising your arm up and rotating your arm. So it seems as though this is a pretty good tool in giving us a good sense of, you know, if you have this much muscle on something that can be done in within 5 minutes, and you've partitioned the right arm, it can give you a decent sense of how much strength your right arm can do. And we see that for the left arm, and we can see that also for the legs. Another tool that we were trying to look at was the amount of walking people could do. And so this is, once again, we're looking at the legs, and the percentage of the lean body mass is on the x-axis. So the more muscle you have, the more you are going towards the 1 on the x-axis. And you can see that in the legs, the more muscle and comparison to the total amount of bone and fat you have, the further you can walk in distance. In distance of 6 minutes, we ask you to go back and forth in a hallway. And you can see that the more this tool is able to determine that. So look, this is another biomarker in terms of looking at it at the muscle and at the basically a group of muscle level. You can see that it can correlate with function and correlate with what you need to do with that arm, what you need to do with your legs. So in terms of this part, it seems as though the DEXA scans in patients are fairly symmetric measure in the upper and lower extremity. And the percentage of lean muscle mass in the legs is moderately correlated with functional outcome measures in the legs, so like walking that we talked about. And then when we looked at patients over about 12 months change and looking at this, we didn't see any significant lean muscle changes. I think that like this is a great tool because it is really easy to do, and it's really pretty easy to analyze. And I think for small phase 2 studies, so it's like small studies that you're just looking at safety and looking at whether there's a change over time of muscle and whether there's stabilization or maybe potentially increase, this could be a really good tool to think about. The next thing that I really want to talk quickly about is this muscle electric impedance myography, which I just realized it's redundant because the term in myography is basically measuring muscle. What this tool is doing is, what I found out is my gym has it, is basically this was done in a lot of times at the mall, and you go stand on something and it sends you a little bit of electricity through your body. And depending on the electrical, the amount of electricity that's able to go through, it can tell you a sense of how much fat and how much muscle you have. And so this was adapted to look at individual muscles. And so it's been a tool that Dr. Statlin was a big proponent of in looking at it in the Resolve clinical trial. And we looked at it in the Wellstone Bilat study and looking at it really in terms of how it compares to the imaging biomarker, the MRI. And so the Bilat study was a study that looked at things over a year, and then now we're looking at it over 3 to 4 years. And what it does is the following. And so it's really looking at in terms of the scale that I like to think about it, like here down here at the lowest bar is really what's going on molecularly, and that changes it at the muscle cell component. And then this is the composition of the whole muscle, and this is the function. We're really looking for the muscle MRI, really looking at it at the muscle. And so what it does is that it's measuring the volume conduction properties. So it turns out that muscle is a pretty good conductor of electricity because it's a big long tube. And so if you put in a little bit of electricity, it will go through pretty well. If the muscle gets damaged or it gets replaced with fat cells, what happens is that there's not as much electricity that goes through, and so you'll have less electricity that will come back. And so here what we're doing, and some people in this room have experienced this, it's a piece of plastic that gets applied onto you, and it's a pretty painless process that puts in a little bit of electricity through you. And so it puts in the electricity, and then it measures how much electricity is coming through it. And that's really what this EIM is measuring. And so we were comparing it to the amount of muscle and amount of fat that we've been talking about in the first half of this afternoon in terms of muscle MRI, and looking at it at the Wellstone study. And so what we found here was that if you looked at the EIM on the x-axis, the more muscle that you have, and that's going towards your right, you can see that it represents on the y-axis more muscle that you measure through the MRI. And this is looking at the tibialis anterior, so the muscle in your leg here that raises up your foot. So the amount of measurement that you get was this really like 3 to 5 minute process can give you a decent sense of how much muscle is in it. And when you can kind of confirm that was when we were able to use the similar AI segmentation process to measure the amount of muscle in the MRI. And so what about looking at it at the, more like at the patient level, and looking at how much that muscle can exert and how much that muscle can push against a skill to give you a certain amount of strength. And so it's the amount of strength that you can flex your foot. And this was a pretty decent test in terms of the amount of electricity that was able to come back. The more that came back was able to be correlated was your strength of it. Had a pretty reasonable correlation. The higher the number, the better it is. So it seemed as though for this electric impedance myography, it was positively correlated with the strength measured by the quantitative muscle testing. And also at multiple frequencies, it was the inverse of the amount of fat fraction in the muscle measured by MRI. And you can also see that with the looking at it with some of the cellular and molecular biomarkers as well, for looking at how much fat and how much muscle are in each of them. So lastly, I just want to thank our patients and a large group of physical therapy evaluators at multiple sites throughout this nation and the world, and also our funding, which includes the Friends of FSH Research. And these are the people that helped out with the analysis here at University of Washington and throughout the Wellstone network. Thanks.
All right, haven't seen anything online. Are there any questions in the room?
Leo, thanks for the talk. And I really do appreciate using these simpler measurements, which can be done more consistently, more frequently with patients. I was curious if you were able to correlate, especially the myography. You did the correlation with fat fraction with MRI. And maybe this is a question for Silvia also, you're able to correlate also the difference in predictivity or predictiveness for muscle function, because you're both looking at walking or foot, muscle strength, and if those match up as well in those correlations.
So you're asking if it correlated with MRI fat fraction and lean muscle volume and really correlated with function. Yeah.
You're both trying to answer the same question, right? Wondering how close.
Yeah, so yeah. Is this application of electricity and how much electricity comes back, does that correlate well with the amount of muscle and MRI? That was the first question. And it was pretty decent in terms of how much electricity was obtained back. And just giving you a sense of how healthy the muscle is was pretty decently correlated with lean muscle volume. That's the first part. And the second question is, what about for function? And what we are measuring with these are really more single muscles. And so and like what Silvia was saying, that a lot of times a function of a muscle is through multiple muscles together. And so the one that I was showing at was really the muscle of the leg, raising it up. And when you look at that strength, when you just basically push against the foot, see how much strength this comes through, you can see that there is a moderate correlation between the amount of EIM measurement versus the amount of strength that you can exert. So pretty moderate correlation.
Just to kind of follow that, what's the resolution? Because I mean, when you grab the thing in the model, it's going all the way.
Yeah, so that's kind of a moving target because these tools are getting better all the time. And so the measurement that we have was a sensor this big, and it's really looking at a small area of that. And so
a single muscle.
yeah, so you can transfer it to one muscle, you can transfer it to a group of muscle, but the issue still is that it's not covering the whole muscle. And so you're still having a sampling error. And the question is, if you're sampling at a different part of the muscle at different times, how reproducible is that? And that probably, it probably depends. For the most part, it's sampling a pretty large area. And so probably depending on where you are, it's probably going to be similar from time to time. And you can use landmarks to decide at what point you want to measure it. But, like kind of the data that Lara was talking about, even within a muscle, there seems to be a lot of variability of that. And so if you're just changing it a little bit different in those kinds of muscles where the problem is at the edges and you move it a little bit further, how much will that change? And I think that that's tough to say, but for the resolution that we're doing now, it seems pretty good. There's going to be a paper coming out that looks at this electric impedance myography on a larger number of patients, and it seems to do pretty well. And so, maybe it won't give us the resolution of the MRI and the just the number of muscles of the MRI, but I think that you can give us it's another way of measuring the amount of muscle and trying to track it over time.
Thanks, Silvia. Okay, so I just want to introduce Glenn Walter and Krista Vandenborne from Florida. The secret thing I can say about both of them is I kind of have like a scientist crush on Glenn and Krista. I first met them probably in person, 10 years ago, maybe more. And, I'm an MR. I did MR science for my PhD. And in the MR world, you're always kind of comparing like who has a bigger field strength magnet. And, and like we had a 1.5 when I did my PhD. And then we kind of relate to the 3 Tesla gains when I did my postdoc. And then we never really got above 3 Tesla, but Glenn and Krista did. And so I've always just been really impressed. Their technical expertise is amazing. Their systemic programs that they built are amazing. And so I think thank you both so much for talking today. And I think the lessons from DMD are really good things to think about in FSHD. And so we're super appreciative of you being here.
Well, Steph, thank you so much for the invite, and it's really quite amazing, and for the Friends of FSH also too for hosting us. It's really amazing to see the amount of progress that you have made since I think it was the first Wellstone study you talked about earlier. We had the group meeting out in Seattle. It's really exciting to see. So what we wanted to do is really just give you a couple examples of many of the different imaging modalities and techniques that you heard earlier, and how we've used that in Duchenne muscular dystrophy. And we're really quite excited because, as you'll see in many of these, and you've been hearing in a lot of the different applications, this is something we took from preclinical models to natural history studies. And we're very proud to be part of 2 approved drugs over the last year that have used MRI methods in their clinical trials. And so we wanted to give you a feel for how that's come along. Here's our disclosures. So, we really started this about 30 years ago at the University of Pennsylvania. And like most people at that time working in muscular dystrophy, we started out with a mouse model. And we really wanted to track the progress or the progression or the digression of the structural defect. The apoptosis, necrosis occurs, the inflammation, which you've already heard a lot about, is a replacement of contractile tissue, but what we also heard about by non-contractile tissue of fat and fibrosis. So what we've really focused on is a number of different quantitative measurements. So I'll tell you a bit about transverse relaxation time, or T2, today. This is the quantitative measurement of STIR that you heard about earlier, as well as, I won't talk about today, some of the bioenergetic differences that we're seeing in limb-girdle muscular dystrophy as well as in Duchenne muscular dystrophy. We can look at fiber remodeling using this T2, as well as diffusion tensor imaging. But really where we've ended up focusing, as being a basic muscle biologist, is really wanted to get into looking at the study of muscle, but really in the human condition, especially the dystrophinopathies, what we've found is it's really a disease of replacement by fatty tissue. And so really where we focused a lot of the clinical trials are in terms of looking at the replacement or of the muscle by fatty tissues and non-contractile tissue. And this is using a method called Dixon, and I'll explain that a little bit later, as well as spectroscopy, which I'll give you some examples. So at the University of Florida, we've really developed a comprehensive MR evaluation of both the boys and adults and female carriers who are lacking dystrophin. And this is an example of a whole body image. During this session, we also look at cardiac function. We move down and we look at the pulmonary function by using, in the subsequent images that I'll show you, these red and green images. The red areas on the image are areas of muscle and water, and the green areas are the areas of just fat. And you can see a healthy control boy here. If we look through the thorax, here's the heart and here's the pleural cavity, versus a boy with Duchenne muscular dystrophy in terms of the large amount of fatty tissue deposition around the body and the replacement of muscles. Similar to the cardiac measurements, we've expanded this to look at pulmonary function, because you can imagine both cardiac and pulmonary function are not only impacted by the absence of the muscle protein, but also by all this accessory fat that's around the body. We heard a little bit earlier about bone density. A lot of the boys and even adults that we look at are treated with chronic glucocorticoids, and I'll tell you about the positive effects. But some of the negative effects of that and the lack of being weight-bearing is that the bones also start to break down. And we're looking at MRI to look at the measurements of stiffness calculated from the images related in the boys. And finally, what we'll spend most of the time focused on today is by using MRI to track the progression of muscular dystrophy, again, where the green is the fat and the red is the muscle. And also we heard about, and we've really been pursuing this quite extensively recently, is tying these changes in MRI characteristics to what we can measure in the blood, if that's proteins, metabolites, or even just basic DNA differences. And so trying to look at different orthogonal axes really to predict and track muscle degeneration in Duchenne muscular dystrophy. We've expanded across the dystrophinopathies. For example, we saw earlier T1 weighted image. This is actually what's called a Dixon image. So what's bright here is fat, and this is a healthy adult control where basically we only see fat around the subcutaneous part of the leg, and this is through the thigh. And here's in the bone marrow, inside the bone marrow. This is a boy with Duchenne muscular dystrophy using the same type image, and you can see the extensive replacement of multiple muscles throughout the hamstrings and the quadriceps. This is the more the adult version of where there's partial translation of a dystrophin gene in Becker's muscular dystrophy, but we can still see the atrophy as well as the replacement of muscle by fat. And here's a young girl who's an affected female carrier, in which we can again see muscles replaced by fat, almost to the same extensive rate as Duchenne muscular dystrophy. So the absence of in this case here, actually look really quite hard. You can see this young lady was biopsied and has very little residual dystrophin. So really we focused on, which is a very clear and obvious target, and we've heard a lot about this, is one is the muscle and the muscle replacement by fat. Moving on to, we've expanded into other types of dystrophies in terms of, and this is an example of those same type Dixon images where this is a highly functional patient, patient living with limb-girdle 2A, which is a calpainopathy, so their muscles lack calpain 3. This is a highly functional patient. This is an example of an ambulatory patient, but interesting, when you look at this image, it's some similarities that you see to FSHD in terms of the replacement of this head of the gastrocnemius muscle by fat, where there's the preservation of the other gastrocnemius. But even that heterogeneity that we heard earlier from Silvia is in terms of, in this case, in calpainopathy, you can have one side of the soleus muscle that's replaced by fat and not the other side, whereas the TA muscle is maintained throughout most of the disease. And we can quantify that in terms of the fat fraction, which you've heard a lot about earlier. One thing that you heard about earlier in a different a way of saying it is looking at STIR positivity. We've spent a lot of times in terms of developing imaging sequences which take the STIR positive or negative and actually calculate what the biophysical property that results in that contrast. The importance of that is similar to fat fraction, where you can go to different scanners and calculate how much fat is occupying a pixel. In this case here, we can go across different scanners or follow the same person longitudinally and look at what is the water T2, which would be a quantitative measurement of the amount of STIR positivity, basically. And we can look at that across different muscles. Here's just an example of, and it's actually maybe more of even a discussion point, is if we look at adult controls, they have a very low STIR positivity, as you saw earlier, or T2. We look at patients with limb girdle, it's elevated in some, not all, in the TA. Becker's muscular dystrophy, again, that adult form of the dystrophinopathy, it's elevated. And interesting enough, in a disease where we don't think there's a lot of inflammation in the TA in terms of myotonic dystrophy type 1, but we also already see an elevated increase in that water T2. If we look in those areas of higher fatty tissue deposition, so we saw earlier right here in the soleus muscle, you can see that water T2 value is actually kind of a dichotomy between what looks to be normal, but actually is decreased in the soleus muscle of people with limb-girdle muscular dystrophy. I bring this up because you'll see in a second is that's a more challenging type of disease to track because the disease progression is relatively slow and more seems to be more similar to FSHD in that case. We had the good fortune of looking into a pediatric disease in which the disease rate is quite progressive and fast. We have currently 228 boys with Duchenne muscular dystrophy in our study, and 150 of them have now completed their 12 years follow-up. This is an example of a partial time lapse of one of the boys that's been in our study now, I believe, for 15 years. And what's really important about these measurements is they're quantitative. So every pixel that we heard about earlier in these images can be represented as a fat fraction, but we can really study the progression of the disease year after year after year. And the patients can come back and we follow them. And from this information, we can calculate their trajectories. So for instance, here, this is a young boy, 7.5 years of age, where we detect a little bit of fatty tissue replacement here in the lateral gastroc. Over the years, you can start to see it progresses, but certainly when you get out to about 10.5 years, almost half that lateral gastroc's replaced. At 11.5, all of it's replaced, and now the medial gastroc's going. And when that boy becomes non-ambulatory, you see that most of the muscles within that child's leg are replaced, except for the tibialis posterior. So Duchenne, in this case, and some other pediatric dystrophies are very fast moving, and it's easy to calculate or visually see the differences. When moving into adult onset dystrophies, it becomes a little bit more difficult. Here's an example of baseline and 5 years afterwards of two people living without calpain 3, so the limb girdle 2A, in which there's very subtle differences in this one gentleman who was actually non-ambulatory over those 5 years. And this young lady here who is ambulatory, if you look carefully, you can see the baseline versus 5 years later, the peroneal muscle groups shows the selective replacement of that muscle. So in this case, in the case of Duchenne, where we see that changes can range from 3 to 8% per year, in most of the limb girdles, we're talking, and also possibly in Becker's muscular dystrophy, about 1% or 2 with years. So it's still a bit more challenging when the disease progression is slower. The good thing is again, with Duchenne muscular dystrophy is there's strong rationale and use of functional correlates. If we heard the importance of moving the MR biomarkers to be related to clinically meaningful function. So in this case, we've shown good relationships between the fat fraction within the one quadriceps muscle, the vastus lateralis, versus 10-meter walk-run velocity times. You can see across the boys the relationship between vastus lateralis and fat fraction. And most of the functional measurements, 4 of them shown here in terms of stair climbing times, stand up and go, a 6-minute walk test, have shown high correlations between the MR fat fraction measurements and the functional measurements. Even more so is the ability to be able to predict future functional declines or the relationship to the ability to do a function. So for instance, here, if we look at vastus lateralis fat fraction versus the ability to perform skills, you can see it in this Kaplan-Meier type curve. You can see clearly the relationship between the vastus lateralis fat fraction and the loss of the ability to be able to perform a task. And what makes sense here is for the ones that require the most muscle force in terms of like supine to stand, you see that's the function that would lose earliest in terms of fat fraction. And when looking at logistic regression, fat fraction does a tremendously good job of predicting future loss of function. Moving into, again, is the adult muscular dystrophies. In this case here, Becker's muscular dystrophy. The relationship is a little bit more difficult, and we start to think about composite measurements. One composite measurement in this case here is the North Star Ambulatory Assessment for dystrophinopathies, which is broken up into a number of different skills. So looking at each one of these different type skill is then rated. And we can see that there's also in this slower moving disease, a relationship in this case here, since the more the adult onset ones seem to involve more muscle groups than Duchenne, where it's all progressing very quickly, but muscles are progressing at a very predictable rate. In this case, we think about taking more composite measurements. So this is work presented at World Muscle Society last year in October by Kelly Rock, a postdoc in the lab, looking at a composite between the glutes and the thigh muscle fat fraction and the relationship between the cutoff of that the cutoff from the fat fraction value and the ability to do a functional task. And what makes sense here is the most demanding ones of muscle force will be the cutoff values are very low fat fractions, for instance, 27%. But tasks such as standing, where they can use other muscles to stabilize, you can see that they can actually still occur to very high fat fractions. And so the composite score, what you see in the case of in this composite one, the composite functional versus the composite MR scores, see a very high correlated and significant difference between the standard for measuring function and MR and these adult dystrophies. Moving on to therapeutics, so this is really important to show, and you heard earlier was that you can visualize the differences in the muscle degeneration, but can you see therapeutic impact? We were lucky that in Duchenne muscular dystrophy that it hits at a point where it's become accepted that the use of chronic glucocorticoid would prolong the ability to ambulate for 5 to 7 years. So we had the opportunity to look in young Duchenne boys, if boys who were not on steroids, or those boys who were on steroids. So if we look at the annual change of the fat fraction within the muscles, we see those boys who are not on steroids, a large increase in both a postural muscle, the soleus muscle, and a high force generating muscle like the vastus lateralis. And this is over 1 year in terms of muscle composition. Really quite amazing that even if this is at 6 months, but even within 3 months using imaging MRI T2, and in this case here, this measurement is a combination of inflammation and fat deposition, is that you can see those boys who remained off steroids, there was an increase in the amount of fat and inflammation in their muscles over as early detected as 30 months, but here in 6 months. But those who are on corticosteroids, we see a drop in their inflammation. And I'll come back to this in terms of a recent clinical trial. So if, again, is that with all the data we have, we're able to model the trajectory of the differences in fat fraction versus age. So for instance, in this case here, these are boys who the soleus fat fraction of age versus fat fraction here. And we can see in white, these are boys who are on steroids, and on red are boys who are not on steroids. The important point of this is by looking at this MR trajectory, we can see the difference between those boys who are on versus off in terms of 50% of their muscle would be replaced by fat, and boys who are not on steroids approximately 5 years earlier than those boys who are on steroids. And so a way that you track their disease progression using these trajectory modeling. But really what's important and what's moved us into the approval stage, FDA approval stage, is upholding to good clinical trial status and really taking the time. So the studies I just presented, most of those were over 2 to 3 sites, 1 to 3 sites. In this case, when you want to move into phase 3 clinical trials, going to have to be able to pull participants from multiple sites. So to be able to do that, if you're going to move to 20 or 30 different MR imaging sites, you really need to follow the MR guidance that the FDA has put out in terms of good clinical trials procedures, which include standardization, constant review of the data coming in, and really fast feedback and compliance with clinical trial protocol, and to make sure that the data is being acquired and the data is being acquired correctly in a timely manner. And that's been really important when moving into the clinical trials. So with that behind us, we've had some great successes. I'll just highlight 2 of those. This is an example of a large phase 3 clinical trial, which was done by a telepharmaco looking at an DUX4 inhibitor for boys who have Duchenne muscular dystrophy, a large phase 3 study, in this case here, randomized placebo control. These are the fat fractions, in this case determined by Dixon imaging. Here's an example of a water image from a boy with Duchenne muscular dystrophy. We looked at the fat fractions throughout multiple muscle groups on the boys who received the inhibitor, HDAC inhibitor, versus those on placebos. And you can see that there was a large change between the treated boys and the untreated boys in terms of their mean fat fractions, which was highly significant by MR imaging across all the muscle types within the thigh that we looked at. And this drug was just recently approved, and this is it's quite exciting to us, is that even on the package insert, it describes the MRI studies that went along with the clinical findings. A more exciting aspect is in this age of gene therapy, we've looked at boys who've been treated both off-label and placebo controls for microdystrophin gene therapies treatments. So I'm going to give you an example. This is an example of a 7-year-old boy with Duchenne muscular dystrophy. Again, the green is fat and the red is water. You can see in this boy that in terms of this rectus femoris muscle, it's almost completely replaced by fat, some of the hamstring muscles, and this is the vastus lateralis muscle. And we see some fatty tissue deposition, which appears as green. Another method we use is MR spectroscopy. So basically, we take an NMR spectra to determine the biochemical composition within a large region. And you can see that down here at the same color coding. From that, we get a measurement of the amount of fat and the amount of water. The green is being fat and the amount of water and muscle is being red. So we can see that in that vastus lateralis muscle, that there's a large amount of lipid. You can see dramatic differences if you look at an age-matched boy who's been treated with gene therapy You can see that all the muscles are well intact. There's very little green within those muscles. And this is more quantitatively shown using MR spectroscopy. Again, we see here lots of muscle and very little lipid. And this was very exciting. In this case, using those trajectory models was, I think, even more dramatic in the sense of the trajectory shown here in green are the vastus lateralis fat fractions, mean and 95% confidence levels from DMD boys who are not on steroids. The purple are the trajectories of DMD boys who are on steroids, showing that difference we saw earlier. And if we look in this gray area down here, this is the level of what you would see in healthy control boys. And the boys who have been treated with microdystrophin are indicated here as these red dots. So well and sustained within the control levels. Boys here who are on, and blue dots are boys who have that, again, that have Becker's muscular dystrophy and some partial translation of dystrophin. So clearly differentiating and being well preserved over, in this case, 3 years. And now these boys are out 6 years later and still looking great. To move this into clinical trials, these are results presented by Krista at the World Muscle Society back in October. The results from a double-blind experiment using microdystrophin at the 51st part in terms of the one group was treated with AAV encoding a microdystrophin. The other group was the placebo control. And if you can remember back to what I showed you in terms of steroids, why I showed that figure earlier, we saw a very similar effect from those boys who were treated with a microdystrophin. Their fat fractions in the soleus and vastus lateralis were lower than those who are on placebo controls. And again, looking at the MRI T2 values, we can see a difference, that dichotomy that we saw earlier, similar to it in the steroid-treated boys. Those boys who are on placebo, their MRI T2 increased over the year, where those boys who were treated with a microdystrophin, it actually decreased. So I hope that I've been able to show, and you've seen many examples throughout today in terms of that quantitative MRI can detect early involvement in multiple muscles. We've seen that in multiple different types of muscle dystrophies. Sensitive disease progression, we've seen that again also in different types of muscular dystrophies today, and it's been shown in limb-girdle muscular dystrophies. Associated with clinical measures of function, clearly in Duchenne and Becker's muscular dystrophy, and a number of limb-girdle muscular dystrophies now. We've shown you that with Duchenne muscular dystrophy, that the MR measurements can predict future functional declines. And really what was key in terms of these studies has led us to be able to do FDA phase 3 clinical studies to show that MR measurements are responsive to intervention. Of course, this was a huge team effort, and Krista's online, and she's going to help me answer all those specific questions, I'm sure. Lee Sweeney, which is the co-director of the Imaging NMD, which is the NIH funding for this study. As well as Lee is the PI of our Wellstone study that's brought a lot of the primary technology forward, and the other imaging folks and people who've helped to contribute to this work. And thank you for your attention, and of course, to all the families that spent the last 15 years traveling to us, at least in Florida, where the weather's most of the time okay. So it's not so bad, but 15 years, year after year, it really takes a team effort and dedication on the behalf of the families. And thank you for your attention.
Okay, so next up we have Jeff Statlin. Jeff is our fearless leader of the CTRN and all things adult and soon to be pediatric natural history study. And the exciting fact about Jeff that we learned last night, well, Silvia, we all did it together, which is we had dessert, ice cream, and yuzu cake while we were eating sushi because things came a little bit out of order, and Jeff rolled with it with no problem. So
you all should try it. It's a new thing. All right. So, I mean, we've heard a lot of, I think, really interesting talks about different approaches for biomarkers in FSHD. We heard about what's been done in Duchenne muscular dystrophy. I'm going to try to circle around now and sort of come back to thinking about biomarkers as tools for clinical trials for FSHD. These are my disclosures. It includes funding from Friends of FSH Research, which we greatly have appreciated over the years, as well as my work for a number of companies. So whenever we're considering biomarkers or any other clinical outcomes for trials, you really have to put it into the context of the disease and think about the mechanism of action for the proposed drug. And there's several challenges as we think about FSHD, and I think we've been hearing about them today. One, it's caused by a toxic gain of function of DUX4, but DUX4 has sporadic expression. This is unlike Duchenne muscular dystrophy or limb-girdle dystrophies, which are due to the absence of, membrane-bound proteins in the muscle, which are ubiquitous. It can affect people from birth through old age, which gets at this notion of variability you've been hearing, both in the age of onset and the rates of progression from one time point to the other, but also an asymmetry in a single person's body. On average, we have a chronic and progressive condition. And if you look at this, at our different functional or strength outcomes, the change in a large group of people is about 2% per year. So a challenge when we think about a clinical trial if it's only running for 1 year. There are these extramuscular manifestations that, if they're present, they're quite important to someone, but they're quite rare in the general FSHD adult population, which would be challenges. You think of measuring them as outcomes. We have no current therapies, and the standard of care may actually differ by country. So this brings us to biomarkers, because in a chronic progressive condition that has a lot of variability, you may want another tool that will show you a sign of treatment earlier. So a biomarker by definition is just a measurable characteristic of the body that indicates normal or abnormal biological processes. It can be a tissue like blood, skin, or muscle, but it can be other parameters like MRI, EIM, or ultrasound. But by definition, it's objectively measured and quantifiable. In clinical trials, this is going to differ from our clinical outcomes, which are going to measure how someone feels, functions, or survives. So as we think about the setting of an intervention and you think about the change in your biomarkers, a person may or may not be aware of it. But ultimately, in order to help get drugs approved, you have to put the change in the biomarker into that clinical context of the disease. And that's a challenge that we're facing now with FSHD. When we think about biomarkers, the FDA really sort of tries to emphasize this point of the context of use. There's several different kinds of biomarkers. A lot of people, when they use this term, they're thinking of a monitoring biomarker, so something that changes as the disease changes. But there's several other different kinds. A pharmacodynamic biomarker, by definition, requires a drug to show that it's pharmacodynamic. It's the response to that treatment. There can be a prognostic biomarker. This would be something about the value today is going to predict what's going to happen over the next year or two. An example of this would be an ALS serum neurofilament, which the levels can predict what happens to people over time. And recently there was a drug approved for ALS based on alteration of a prognostic biomarker. When we think about biomarkers across all of them, there's several key concepts to keep in mind as we try to get them ready for clinical trials. Does the biomarker measure the concept of interest? For example, if it's a protein in the blood, it's an assay for measuring creatine kinase, then is it actually measuring creatine kinase, or is it sensitive to other proteins that might be in the blood? If we're talking about MRI and fat fractions, is it really measuring the fat inside the muscle? The reliability, you can think of two aspects of this. One is related to the tool. So if you run the same samples twice on the same assay, are you going to get the same results? So variability in our tool for measurement. But there's also the natural variability in FSHD. If you take samples at two different time points, but nothing's really clinically changed, you need to be aware of both of these as we use these biomarkers. The validity, which typically we're referring to how well this biomarker actually relates to other types of measures for FSHD, in this case, usually strength, function, or patient-reported impact. Responsiveness, this could be responsiveness to FSHD changing over time, or it could be responsiveness to the intervention, the drug and or any other intervention that you could think of doing, like exercise. And then finally, they all sort of circle back to this. You're going to need to have some concept of how large a change is going to be clinically meaningful to a person, because ultimately you're going to be looking at small changes in your biomarker, over shorter periods of time and wanting to infer that if you continued out in time, that would be important to an individual. When we think about MRI as a biomarker, there's several different categories that you're hearing people talk about already today. Certainly as a monitoring biomarker, a way to see the change in the disease over time. This could be pharmacodynamic, but we would need the drug in order to help us understand that. This would be the concepts like lean muscle volume, quantitative fat fraction, or fat infiltration. But we're also hearing people talk about it as a prognostic biomarker, the STIR sequence, the intermediate fat fraction, either predicting individuals who may change more rapidly or as indicators of molecular activity at the level of the inside the muscle. So when we think about MRI fat fraction, does it measure the concept of interest? One way you could look at this is to correlate the different ways of quantifying the fat in the muscle. There's magnetic resonance spectroscopy, which we just heard about. It gives you a nice peak for the fat and the water signal coming from a region of interest, and you can use that to calculate the fat fraction. And indeed, our other methods for calculating the fat fraction with MRI, like using the Dixon technique, have a very high correlation to the MRS fat fraction measurement. You can also look at a correlation between the fat fraction as measured by MRI and the pathology and histology from a sample taken from the muscle. In this case, it's a moderate correlation, but of course, you're not just looking at fat when you take a piece of muscle, you're looking at all the other pathological changes that are going on, but does argue for this being a surrogate for the disease state inside the muscle. We just heard a fantastic talk from the work that Imaging DMD has been doing about its use in Duchenne muscular dystrophy. But I think, there's several aspects about how they've translated the use of this biomarker to understand its context in the disease that we're going to need to grapple with in FSHD, which has perhaps more challenges. As he showed you, they followed boys for a long period of time. This enabled them to get these really useful curves of the change in the fat fraction versus the age of the boys. You can see it's very characteristic in Duchenne muscular dystrophy. In the vastus, they tend to have about 50% replacement at about 12.5 years of age. In the soleus, it's a little bit older, so a later muscle in that disease. But this means if you have the boy, his age and his baseline fat fraction, you can make estimates about what you would expect that to change at in 2 or 3 years. They also followed these boys long enough to show that association between the fat fraction and function, in this case, using the 6-minute walk test in the bottom left. You can see as the fat fraction on the x-axis is increasing, their distance on the 6-minute walk test is dropping, and at a certain point, they just lose the ability to do the task. Because they're losing that ability and we're following over time, you can now create these probability curves, which you see on the bottom right of the screen. If you look at the red line, that's the probability of losing the ability to do the task, the 6-minute walk test in this case, going up as the fat fraction increases. But this gives you the ability now to start talking about whether a change, if you see it in a trial, is going to be clinically meaningful and how. A change of 20% in the fat fraction may reduce your chance of losing ambulation by 20%. You can start to make that connection. When we think about MRI and FSHD, the earlier studies really used a similar technique in a way to what's being done in Duchenne. They took the midpoint of the muscle, took several slices, showed you can, figure out which muscles are there and how much fat is in each muscle. That's what you see on the top. But FSHD has a lot of heterogeneity, and there's not the same characteristic effectiveness of an individual muscle versus the age of someone with FSHD. So we've moved to a different type of technique, a whole body technique that Silvia talked about earlier. They use AI to help segment the muscles and analyze them, but this gives you the whole muscle and all of the muscles in a region that you can analyze over time. So we're doing the technique. The next question really is it a reliable thing to be doing? When looking at quantitative MRI in general and FSHD, when reported reliability to the disease at 2 points in time that are close together has been quite high. You can see this image from a study using Springbok's technique showed the fat fraction between baseline and 3 months, a time when really not much would be changing in an individual, and you can see that variability is quite low. So within the range of what would be useful in the setting of a clinical trial, around 3%. Does quantitative MRI relate to other things that are of interest in FSHD? In this case, severity, strength, and function. And you can see in the bottom left, this is the Dutch group. They took an average of the quantitative fat across multiple muscles in the lower extremity. They showed moderate to strong correlations to a severity score, to the individual muscle strength, as well as to a combined measure of function called the motor function measure. On the right side, you see data from the Fulcrum DUX4 trial, and you can see a correlation to an ambulatory measure in the top 2 panels or to the reachable workspace in the bottom 2 panels. And I think the point I want to make here is those correlations do depend on the muscles that you're including. For an ambulatory measure, you really need to be thinking about the muscles of the leg. For the reachable workspace, you would need to be comparing it against the MRI of the muscles in the shoulder girdle. As we think about change over time, it's much more complicated in FSHD, as we've already mentioned. Overall, the longest study we've had using quantitative MRI is reported on 5 years of follow-up. If they averaged over 19 muscles in the legs, they saw a 2% change over 5 years. If you look at our other measures of strength or function collected in that study, the same relative change at that time period was 8%, so larger than what they saw on MRI, which becomes a challenge when you think of this as a biomarker in the setting of a clinical trial. So in order to use it, you need to ask, are there some sets of muscles that maybe are changing more rapidly? And indeed, you've been hearing about this today. You can use several features of the muscle at baseline to predict a larger change over 1 or 2 years. If you look in the top panel, the change in the fat fraction is on the y-axis. Those blue dotted lines would be no change. I apologize, they're slightly shifted in this presentation. And then on the x-axis in the top, it's the fat fraction at baseline. You can see that intermediate fat fraction muscles at baseline had much larger changes over this time period. You can also look at the number of STIR-positive muscles. That's in the middle panel. And you can see the more STIR-positive muscles that were present, the larger the change in that fat fraction over 5 years. But you can also look at a simple severity score when someone comes in at baseline and see that individuals who would have changes already in their legs based on that severity score have larger changes in the follow-up period. And so as we think about the use of quantitative MRI in clinical trials, we're likely going to need to use subgroups of individuals that we'll be following over time to have it behave more like a biomarker. The other way we think about DUX4 activity is its ability to select for muscles that have molecular activity. So that STIR signal and those intermediate fat fraction muscles also appear to select for individuals who are going to show you those DUX4 related targets on muscle biopsy. The bottom right, this is from Dr. Tapscott's Wellstone 2 BILAT study. And you can see the fat infiltrations along the x-axis, the DUX4 composite is along your y-axis. And as you hit 20% fat infiltration, you see a large increase in the level of that DUX4 score, as well as those stir-positive muscles showing you. So arguing that MRI may also have potential as a prognostic biomarker in these patients. So if we think about using it as an outcome measure, does it provide a sample savings if we're thinking about planning a clinical trial? You can take the placebo data from the recently completed Fulcrum Reach study. They used AMRA, and they looked at a quality of MRI called the muscle fat infiltration, and they looked at those intermediate muscles as a composite. If you take the variability they saw in their study and you planned for a study seeing a 2.5% difference between your groups, you would need 85% per group to see that. To put this into some kind of perspective, using something like strength, if you wanted to see a 2.5% difference between groups, you would need double that number, 160 per group. Obviously, if you're going to see a larger change, your number drops considerably. So as we're thinking about MRI as a biomarker in FSHD, there's still some pieces that we need to fill in, although we've done a lot of the work. We need better methodology for predicting which muscles are going to change over the time frame included in trials, 1 to 2 years. The ability to tie these changes to future functional motor performance or outcomes is going to be particularly important, and we heard Silvia talking about that earlier. Can the field settle on the technique or techniques of highest interest? Remember, though, while the sensitivity of a given technique may not be better than a strength or functional measure, it may still respond better to a treatment once we have one. So it's something just to keep in mind. And then can we use the signals on MRI for other things like the inclusion criteria or prognostically in the setting of a clinical trial? And indeed, it looks like there's mounting evidence that we would be able to do that. So as we think about future directions now for the field, there are several ongoing studies, some funded by Friends of FSH Research. We have our MOVE Plus study, which is charting the connections between MRI, muscle biomarkers, blood-based biomarkers, and strength and functional changes. We're just over halfway enrolled in that study. And so we'll be able to answer many of these questions as that cohort finishes up. There are the ongoing clinical trials, like the Avidity trial, where they've identified this DUX4 related circulating biomarker that they have yet to announce what that is. But again, the ability to have a treatment in an active trial will teach us a lot about our biomarkers and what may or may not be important in terms of changes. Will the regulatory authorities consider these biomarkers for accelerated or early approval of drugs? While the FDA seems open to the idea, you have to be able to make the argument that the biomarker reflects a key pathological process in the disease and that a change in that biomarker now is reasonably likely to predict something important to people in the future. And the same type of work that's going on in our adults needs to really be happening as well in the pediatric population. And as you heard earlier, we have a study that's been funded by the NIH that's starting in the next couple months that will be measuring these different outcomes in children. All right, thank you very much.
I guess real quick, at the end of that, you're talking about FDA approvals and kind of the need to reasonably prove that using biomarkers has a downstream effect of functionality. Has anybody, if you were to say take an improvement that is a stage 1 or stage 2 of her predictive AI model, you know, it says this down the road will create functional improvement, even if you haven't been able to prove that. Has anybody done that in any other areas, do you know, and gotten it to meet that reasonability standard?
Well, I mean, I think it's, you have to be able to tie the connection first before you could talk about using the model, I think, to suggest it for an individual who's maybe at a different point in their progression. I do think there's several ways the model will be very helpful, though. You know, you can talk about historical control data and what's called propensity matching, where you sort of create these virtual placebo groups that you can compare against. And you would need to have those models for that. I don't know of a drug that's been approved based on a models, an output like that.
Yeah, I think there are examples in like outside of neuromuscular disease. I think even I think in diabetes and in cancer as well, we're using machine learning models to essentially guide clinical trials. It's really common in heterogeneous diseases, pretty much. Like that's really the time when it's really valuable, and combined with when it's somewhat rare, because the more heterogeneous in theory, statistics wise, if you want to account for that in your design, you just need more people because of the variability. So that's where it really comes. So there are examples, and I think the FDA is moving to more accepting more machine learning and AI. And there's obviously a lot of rigor in terms of evaluating the validity of those techniques. But yeah.
Any other questions?
Krista, can you hear the questions? Can you give me a thumbs up? Okay, great.
Can you guys hear me here? So, I've been Sylvia, I missed your presentation just now. I'm from Canada. I had to check out what the hell's happening to us up there. But anyway, I've seen it before, and it's a really good correlation of all of the, the muscles and so forth across a lot of FSHD patients. But that's looking at all the different muscles. Is there a single muscle? Let's say you looked at all the biceps of all the people that have FSHD. Would there be something to that, in terms of looking at the progression of FSHD? Because the muscles are so higgledy-piggledy, right? One side stronger than the other, and different patients have different effects. So is there a muscle that would be a good tell across all FSHD patients? No? Oh, damn.
I mean, I think, can I just make one comment? I mean, I think, yeah.
I would like to look across all the biceps or something like that.
Well, I mean,
that's what
the
we do.
semimembranosus is commonly affected.
I know, but it doesn't distinguish
It
patients.
does not, yeah.
No,
I mean,
it's everybody's.
I think one of the things that we're excited about, and granted, we're heavily in like let's make more models, we're in that kind of realm. But I mean, I'm still very excited about if you have, 100 patients that come into your trial and 10 of them have shoulder girdle features that are changing, we should do a shoulder task in those patients, or have that be a heavily weighted reporter compared to a patient that has a glute and a thigh, muscles that are vulnerable. So I do think the cool thing about these models is they're not really data averse. So we could put, composite measures like the things that, in DMD and in FSHD we've been doing, where there's a lot of tasks in the model, and try and predict your current state and where you're going to progress, both in an imaging perspective and in a functional perspective. And so I think once we can kind of demonstrate that works, it's a little easier to think about, well, how sparse can we make those, those data models? But at this point, I think it's so exciting that we can see the heterogeneity. We can start to think about that. And then I think we can go the other direction, which is how simple, if we could use DEXA, that would be great, but we're not quite there yet.
Thanks.
I'm officially replacing the word heterogeneity with higgledy-piggledy.
Yeah, I know.
I'm just wondering with the higgledy-piggledy that we've been speaking about for like 20 years, with these type of digital twin models, can we start to envision a world where you can enroll a patient with a shoulder, with a shoulder sort of improvement regimen they have to go on versus a glute, and put them into the same trial, and perhaps even think about instead of systemic therapies, more localized therapies under one blanket? Is that something that still people think is not possible?
Who wants to take that one? Stephen, you're the systemic guy.
Yeah.
Jeff?
Well, I mean, there's been discussion about this for a very long time, especially for rare diseases. Can you take different outcomes, that are catered to the individual who's coming in the trial, all of the outcomes which have been sort of independently validated, and create a trial based on sort of the composite, where each individual's success or failure would be based on something maybe a little different? I think it's possible to do that. You would have to have each of the elements, though, of the combination that you were using sort of separately, validated in the field. But I don't know an example, again, of a drug that was approved based on that approach.
I mean, I think the other way to think about this is, like, right now, what's used is really a model. The model is saying that the placebos are what would have happened in the treatment group if they didn't get treatment, right? Like that is the current version of the model, which one could argue is not as accurate as a model that represents the heterogeneity of the disease. So just I mean, not to say that that is the way clinical trials happen, but it's a good way to kind of conceptualize the issue.
Okay, we'll do one more question because we have to use this format.
I just had a question for Glenn and Krista. It was interesting about Duchenne's and when you talked about the drug that was given to them and they were able to decrease their fatty infiltration. Does that mean the muscle that was gone regrew, or what happened?
Glenn, do you want me to take it?
Oh, okay. Were you guys able to hear that?
Yes.
Okay.
So I can address it. I think that we talked about two components. Glenn presented two components. We looked at the changes in fat fraction, so the change in fat fraction over the course of 1 year. And then he also presented the T2 values. So typically when we look at fat fraction in muscular dystrophy, there's really only one direction that we've seen so far, which is an increase in fat fraction. And what we measure is the difference in the amount of change that happens. When we look at T2, which is a combination of really muscle inflammation and fat fraction, we do see a decrease in T2, but that's probably more driven by a decrease in inflammation or maybe a decrease in loss of sarcolemmal integrity. We have looked many times to see if we can see actually an increase in muscle, in a decrease in fat fraction. I think if you do, then it's probably because there is an increase in muscle mass rather than a decrease in fat fraction. That's just speculative, but that's kind of our assumption.
Can I say just one more thing?
You have to answer.
Oh, what if it's real quick? I think one of the things that I've been most excited about with these data-driven models is that we do kind of see different results of different predictive models if you look at fat versus looking at lean muscle. And so I think the example I've just kind of tried to make sense of is like, if you can't lift your toe, if you have foot drop, you walk differently. And so those muscles that are going to compensate will probably get bigger. They probably will hypertrophy. So it makes sense that you don't always have to have this kind of simple model of the muscle is this big and it just gets replaced by fat and it stays that size. So I do think that we're going to see examples of some muscles atrophy across their course, some muscles hypertrophy, and it's more complicated. But being able to model those at the same time will be really helpful to understand the disease. Well, I just want to thank everyone for staying late and for your attention, and for everyone, all the speakers. Thank you very much for your expertise, and we really appreciate it. So on to not angering kids.
Let's also thank Seth and Premi for the work that you're doing for months to put together this program. So thanks everyone for participating.
Thank you.